Hello and welcome to this part of the educational workshop. We'll talk about models of communication in brain networks. Our jumping off point is this idea that the connectome is really a map that tells us how easily any two areas could communicate with one another. The anatomical connectivity of the brain really supports a myriad of elementary signaling processes in the form of electrical impulses, and the superposition of many of these communication processes ultimately manifests as patterns of interregional covariance or functional connectivity. So the two domains here, structural connectivity and functional connectivity, are empirically accessible to us. Communication is something that we must infer. So what we'll talk about today are really three questions related to the nature of this communication process. That is, what types of communication processes are biologically plausible? What types are suited to the underlying architecture of the connectome? And what types are actually empirically supported? One way to think of communication is really as lying in a spectrum, the one that ranges from simple diffusion-like mechanisms all the way to highly centralized routing-like mechanisms. So on this side, for instance, you might have something like shortest path routing, where you have a source node that transmits a signal that takes the shortest set of contiguous edges to get to some desired target. On the other hand, you might have something like a random walk, where a source node will transmit a signal, but that signal will be randomly forwarded among the neighbors until it reaches its desired target. And interposed between these two extremes are a number of other communication mechanisms that have been proposed by various authors that all envision communication taking place slightly differently. But probably the most fundamental of these, and the one that's most logical to start with, is shortest path routing. If we were to think about what is a good communication model for the brain, we'd first have to think about how communication actually takes place at the cellular level. Here, what we know is that action potentials travel down axons across electrochemical synapses, and then are then transmitted to the postsynaptic cell. So a priori, it might be desirable to minimize the number of synapses that signals have to go through. Why? Because signal propagation via electrochemical synapses is likely to be slow. It's a noisy process, and it's one that requires both energy and materials. So energy to depolarize and repolarize the membrane, to package vesicles and so forth. So minimizing the number of synapses is really desirable. And a good candidate then for communication mechanism would be shortest path routing. With shortest path communication, we first have to distinguish between really two concepts, and that is the concept of a walk and the concept of a path. With a walk, we're really talking about any non-repeating sequence of edges between two nodes. So we might have a source node and a target node, and a walk could be something like this, shown in red. So the length of this walk is 5. There are 5 edges that interconnect these two nodes. A path, or a shortest path by comparison, is can be taken between the same two source target nodes, but it is the shortest contiguous set of edges between them. In this case, the length of this path is 3. And it's important to note that, of course, there could be multiple shortest paths between two nodes, for instance, one that goes through these particular nodes. Now, if we assume that signals are forwarded sequentially to one another via shortest paths, we can actually measure a number of important statistics about the importance of individual nodes and other components of the network to communication. So for instance, we can measure something like the closeness centrality of a node, which is the mean path length from a node to all other nodes in the network. So it tells us how well that node can influence its neighbors and other members of the network. We can also compute something called the betweenness centrality of a node. So if we take all possible shortest paths between all pairs of areas, we can ask how many of those shortest paths actually go through a particular node I. If a node sits on many different shortest paths, then it's theoretically well positioned to act as a bridge or as a way station for signal traffic. And we can then extrapolate that kind of thinking to the network as a whole. So we can ask, for instance, what is the characteristic path length of a network? This is the mean path length between all possible pairs of nodes. Or conversely, what is the global efficiency of a network? So the inverse of the characteristic path length. In both of these cases, the idea is that if you have low path length on average in a network, that implies that you have greater efficiency, potentially a greater communication capacity, and lower potential for signal attenuation. And there's a well-developed literature over the past 10, 15 years that has actually shown that shorter path length is associated with greater cognitive capacity, engagement, is concomitant with normal neurodevelopment and so on. So it appears that shorter path lengths are a desirable property of brain networks. What I want to show you in the next slide are really two kind of lines of reasoning for why shortest paths are potentially a desirable property in brain networks. One is that brain networks, as you saw earlier in this workshop, are a canonical example of what are called small world networks. So these are networks that have path lengths that are, by definition, close to the theoretical minimum of rewired random networks. In other words, brain networks have nearly minimal path lengths already. The other piece of evidence comes from what are called minimally rewired networks. So these are generative models in which you can take real connectomes that are shown in blue here. So for instance, it can be from the macaque or C. elegans. You take the nodes from those networks and you place them in their original XYZ locations, but then you start adding connections such that you minimize the total wiring cost of the network. So you're trying to place connections such that they are such that you can recapitulate the density of the original networks, but with the shortest possible set of connections. And when you do that, when you create these minimally rewired networks shown in red, what you can see is that the total wiring length of these networks can actually be reduced significantly. However, this actually comes at a cost, and that is that the path length, so the topological path length in these networks actually shoots up. In other words, it is very costly to maintain short paths in brain networks, but it appears to be a property that's or a cost that's readily incurred in the development of these networks. Now, there are a number of arguments for why shortest path routing might be a desirable communication mechanism for the brain, but that is not really the whole story. When we think about shortest path routing, we're really making two assumptions. One is that the network elements, so in our case, neurons or populations of neurons, actually know the global topology of the network. They actually can compute the shortest path to the desired target node, which is not biologically plausible. The other problem with shortest path routing is the following. So this is a adjacency matrix derived from diffusion-weighted MRI. So it has 1,000 nodes by 1,000 nodes here. This is structural connectivity. And what I'm showing you on the right-hand side here are just the connections that participate in at least one shortest path in this network. What you can see is as you go from this original network, you go from about 24,000 connections. And when you consider the shortest path skeleton, you have only about 6,000 connections. So in other words, if you assume shortest path routing, you're assuming that only a small proportion of the total infrastructure is ever being used, and the rest is vestigial, which does not seem like a good use of the available resources. So there have been a number of studies that have asked the question, well, could communication actually occur along other routes as well? And that brings us back to this kind of diagram. We can actually relax some of the assumptions of shortest path routing just a little bit and consider what are called path ensembles. So with path ensembles, or the so-called k-shortest path framework, what we're asking is, we're not going to focus just on the top one shortest path between a pair of nodes. Instead, we're going to focus on the top two shortest paths or walks between nodes, or the top 5, or the top 10, or the top 100. And these are some examples of that you can see here. So in each of these examples, you have a source node and a target node, and you have the top k paths between them. And what this allows you to do is to say that, well, we're not going to consider just a single communication channel between our two nodes of interest. Instead, we're going to consider multiple communication channels and open up this kind of almost a spindle that allows us to take advantage of many different routes. The immediate advantages of a mechanism like this is, first of all, it'll allow you to access alternative, optimal paths. And in doing so, you're going to have a much more resilient communication process, one that does not depend on just one single communication channel. The other is that it'll implicitly then allow more network connections to contribute to the overall communication process, making it more biologically plausible. And the important thing that you should keep in mind, and this is going to be a recurring theme throughout the talk, is that if we start to consider not just the shortest path, but the top k shortest paths, this will start to drastically change our view of what are the most important nodes and connections. This is an example of that here. What you're seeing are the most central edges and the most central nodes in the network, either when we consider the top one shortest path, shown in teal, or when we consider the top k shortest paths here, shown in orange. What you can see is that suddenly, for instance, a lot of these frontal connections that involve the insulae and so on start to become more important. So your very picture of what are the hubs of your network, what are the most important connections, changes drastically when you start to consider alternative communication mechanisms. We can take this idea of path ensembles further, and we can really generalize it to include or encompass all possible walks between two nodes. And this is really where this measure of communicability comes in, which is a measure of the total communication capacity between two nodes. And the way that it's computed is we take all walks from a source to a target node, and we compute a weighted sum. And the way that we do this weighted sum is we weigh the short walks more. So shorter walks that are more direct or go over fewer synapses are considered more important. And longer walks that are, more extensive polysynaptic pathways are weighted less. So an example of this might be something like this. In black, you see a source node and a target node. And if we want to compute communicability, we take the number of walks of length n, and we divide that by n factorial. So for instance, there is no direct connection between our two nodes. In other words, we go 0 divided by 1 factorial. There is, however, one walk between these two nodes that is of length 2. This is, in fact, our shortest path here. So that it gets added to the sum. If we consider walks of length 3, we find that there are 2 such walks that are shown in green. That gets added to the sum, and so forth. And the reason for using this particular weighting function is that this becomes the Taylor series. So this particular sum can be expressed as the matrix exponential. In other words, it's a very convenient and elegant way to compute these relationships, and it can be done in a single line of code. Now, as we kind of go towards communicability, we're really now talking about all possible communication pathways between two nodes, and that in a way takes us logically to this other extreme of random walks. So with random walks, what we really have is a situation in which we have an agent, or a quote-unquote walker, that probabilistically moves through our graph of interest. So we might place our walker in this particular node, and at any given time, the walker looks around and sees how many other neighbors it has, and it can move to any one of these neighbors with equal probability, in this case 1/4. It may then choose to move to one of the neighbors, and what's really being done as this process goes on is that we have our adjacency matrix that we've converted into a transition probability matrix. So any one of these rows that tells us where the walker is currently sitting is converted into a row of probabilities that all sum to 1 that give us the probability of moving to any one of the neighbors. As the walker moves to a particular neighbor, we simply move down to the corresponding row for that node, and we go through this process all over again. If we allow this process to develop, this random walk to develop, we can actually trace out a trajectory for the walker. And that tells us a lot about how communication can be supported on this network. And in fact, we can compute a number of measures that are analogous to what we had with shortest path routing. So for instance, we can compute the mean first passage time, which is the expected time for a walker to travel from node I to node J. And it tells us how easily nodes I and J can communicate with one another. We can estimate what is the minimum number of walkers that we would need to generate at node I to guarantee that at least one of them reaches node J. This is the so-called resource efficiency, and it's simply a measure of how strongly you need to broadcast your signal in order to guarantee transmission at some particular target node. We can also ask what happens when we have two walkers that start at nodes I and J, and what is the expected time for them to meet on this network, the so-called coalescence time. The nice thing about this kind of framework is that it's infinitely extensible. So we can introduce all kinds of other constraints that are potentially more reflective of the underlying biology. So we can introduce biases where walkers are more likely to spread to some types of neuronal populations, but not to others. We can introduce memory where walkers are unlikely to double back on the path that they've just been on. And we can also introduce interactions if we have multiple walkers that potentially carry multiple signals in this network. Now, that brings us to an interesting question, which is, could random walkers then find the shortest paths in our network? Now, if we have a situation like this, where we have a source node and a target node, and there's only a single unambiguous path between them, then it's obvious the random walker can find this particular path. But what happens in a situation where the random walker actually has to go through a series of nodes that themselves participate in multiple connections and increase the potential for the walker to get lost? This brings us to two interesting statistics called search information and path transitivity. With search information, what we're asking is, what are the degrees of the nodes that a walker encounters on its way from source to target? If a walker has to go through many well-connected hubs, then the potential for the walker to get lost is increased. Conversely, if we can also compute the so-called path transitivity, which is the number of transitive closures along this path. In other words, how many close triangles do we have along this path? The idea is that if there are many close triangles along the path, then even if the walker were to go off the shortest path, it could still get back on the shortest path and eventually reach its desired target. So what the authors here, this is a study led by Joaquin Goni, what the authors did here was to take structural networks in human diffusion-weighted MRI and to compute the search information at the path transitivity between all possible pairs of areas. And they then compared these patterns of search information at path transitivity to resting state functional connectivity in the same participants. The idea being that if these two are good models of how the brain communicates, then you should be able to predict functional connectivity from structural connectivity. What they showed was that indeed there was a relationship. So the greater the search information between a pair of areas, the greater the number of high degree nodes that you encounter along the way, the greater the potential for information to be corrupted or lost in some way along the way, the lower the functional connectivity between those areas. And conversely, the greater the path transitivity between areas, the greater the number of transitive closures and the greater the potential for the signal to be preserved as you go from a source to a target, the greater the functional connectivity between those areas. So this is a very instructive example because first it shows a good paradigm for how to actually make predictions based on these communication models. But it also shows this important point that there's a difference between shortest path routing as a communication mechanism versus shortest paths as a general property that any network has. And in fact, many different communication mechanisms can potentially take advantage of the shortest path infrastructure in our networks. So what the authors then did actually was to ask, well, how well do routing and diffusion compare, and how well do they work on different types of architectures? And what they did was to grow a number of synthetic networks that tried to optimize either shortest path routing or diffusion. What you can do in such a case is shown here. On the y-axis, you have efficiency under routing, and on the x-axis, you have efficiency under diffusion. And here is what's called a morphospace. It's a space of all possible network configurations that can be grown under these constraints. What you can see is that this morphospace actually has multiple fronts that capture very distinct configurations. Now, what you see in front one, for instance, are a number of networks that are better at routing than diffusion. And if you take a look at some of the examples of these networks, you see that many of them actually have either clusters or in some cases modules or communities. And the reason they're better at routing compared to diffusion is that communities by definition trap random walkers. They have lots of connections among the constituent nodes, but relatively few connections to the rest of the network. So random walkers just circulate in these communities. Conversely, on front three, you have a number of networks that are better at diffusion than they are at routing. And in each of these networks, what you can see is that there is a strong connective core where even if you start out somewhere in the periphery, a random walker could very easily access this core and then potentially have a way to get to anywhere else in the periphery. So diffusion is not disadvantaged in these types of networks. Now, as you saw from earlier in this educational workshop, the brain really has both of these canonical properties. It has a prominent community structure, but it also has a connective core or a rich club. So a question for you then at home is, which of these do you think works better for brain networks? Fortunately, the authors of this paper actually address this question. Here they're plotting again routing and diffusion efficiency, and they're plotting it for different naturally occurring and man-made networks. And what you can see is that especially for networks that are derived from the nervous systems of various model organisms, including the human, you see that they lie close to the identity line. In other words, by virtue of the fact that brain networks have both communities as well as a rich club or core, they can support both routing and diffusion well. So they appear to balance routing and diffusion. And this is another, I think, important point to keep in mind, which is that how well a communication mechanism fits a particular network strongly depends on the architecture of that network. There is no single communication mechanism that's optimal for all types of networks. So I just want to close by showing you one exciting new direction, which is this idea of navigation. Now, with routing and diffusion, we have two concepts that are mathematically fundamental, but neither is really completely satisfying as a biological mechanism. The question is, could we envision something that's decentralized, like diffusion, but that takes advantage of the infrastructure in a biologically more realistic way? So one kind of class of communication processes that we can consider there is called navigation. These are communication processes where signals are forwarded between nodes based on some other underlying metric space, but not necessarily knowledge of the network structure per se. So what the authors did here, this is work by Keilholz again and Andrew Zalesky, is they constructed such a navigation process where they where the nodes are forward signals to neighbors that are closest in physical space to the desired target. So the forwarding mechanism here does not rely on knowledge of the network structure, just on the knowledge of the spatial embedding of the network. And what they were able to show was that regardless of how you define the structural networks, when you look at how well this strategy can actually recapitulate the shortest path in the network that's shown in red here, the so-called success ratio, this type of communication process can actually oftentimes recapitulate well above 80% and often close to all of the shortest paths in different types of brain networks. And this is significantly greater than both randomly rewired and randomly repositioned networks. So it appears to be a communication process that works really well for spatially embedded networks like the brain. Now, finally, the question is, when we compute these communication models, what are we getting in practice? Really, what we have are different ways to conceptualize polysynaptic communication between different brain areas. So with, again, work by Keilholz again and Andrew Zalesky, what they did was to ask how well do different communication models actually predict functional connectivity and behavior. So they first looked at functional connectivity. They took structural networks, computed different types of communication models on these structural networks, and then asked how well they predict functional connectivity. And what you can see here is that if you compute these communication models, most of them actually end up outperforming just static structural connectivity as predictors of functional connectivity. In other words, by embodying polysynaptic communication and this kind of long-range neural signaling, they can actually tell us a lot more about functional connectivity than just structural connectivity can on its own. And they're useful transfer function. What they also did was to compute these communication models on the connectives of individual participants and then ask how well they can predict individual differences in behavior. That's what you can see here. This is on the y-axis, the prediction accuracy for behavior, and on the x-axis are different predictors. You can see that the best predictor by far is functional connectivity, and the worst predictor is structural connectivity. But communication models computed on structural connectivity greatly enhance the predictive utility of structural connectivity on its own, and they actually bridge the gap to functional connectivity. So even though they do not necessarily outperform functional connectivity, they add information to the underlying structural connectivity patterns that boosts the predictive utility of this modality. Finally, I just want to point out that in addition to studying the nature of neural signaling, these communication models are also useful in other domains. One is in the domain of brain stimulation. So we know that different stimulation methods, such as transcranial stimulation and so forth, can be used to treat different diseases and also to enhance cognitive performance. But we don't really know how these methods work. So the communication models that I've shown you can be adapted to these kinds of questions, where you can simulate the effect of a particular perturbation in some part of the network, and then you can try to see how that perturbation would affect areas that are connected to the stimulated area, and areas that are connected to those areas, and so forth. So you can really use these communication models to try to understand how network architecture will condition the effect of the stimulation. The other domain where these models are very useful is in modeling the spread of misfolded proteins in neurodegenerative diseases. So we now understand that many neurodegenerative syndromes, such as Alzheimer's, Parkinson's, and so on, have to do with normal proteins misfolding and inducing misfolding in other normal proteins in the brain. And the deposition of these proteins ultimately causes cell death and atrophy, and these proteins appear to spread transneuronally, that is, from cell to cell. So these kinds of communication models can then be readily adapted to try to predict patterns of atrophy in clinical populations, and therefore have uses that extend much beyond just studying communication on its own. So just to summarize, I hope I've convinced you that most network measures assume a specific communication mechanism, and it's important to keep in mind what are the associated assumptions and the biological plausibility and the ability of those communication mechanisms to make predictions about functional connectivity, behavior, and so forth. The shortest paths are a critical substrate for communication, but shortest path routing per se is not the only possible communication mechanism. And in fact, we can really think of many different communication mechanisms that might operate on brain networks. And that these communication mechanisms really depend on the types of structures that you have in your network, such as modules, cores, and so on. And finally, that potentially multiple mechanisms could operate simultaneously. And this whole field of communication really has to take into account very complex trade-offs between the efficiency of a communication mechanism, the costs associated with that mechanism, the types of resilience that it'll confer, and so on and so forth. So thank you very much for your attention. I hope you enjoy the rest of the workshop and enjoy the conference as well.
So today I'd like to present on the topic of modular structure in brain networks. Now, typically when we analyze a brain network, or any network for that matter, we can focus on two extremes. On one hand, we can consider properties of individual nodes and edges, for instance, the number of connections they make, or how central a particular node or edge is with respect to some process, like shortest paths in the network. On the other hand, we can consider the opposite extreme, where we take the network as a whole and make measurements of, for instance, its average path length between nodes, or the efficiency of shortest paths. And these two extremes comprise local and global scales, respectively. But situated between those two extremes is a rich mesoscale, where we consider not properties of individual nodes or the network as a whole, but of collections of nodes. We call these modules, communities, sometimes even clusters or subgraphs. And when we describe a set of communities that divide the network into different clusters, we consider them a partition. Why should we study this type of structure? Why should we study this middle ground? Well, on one hand, focusing on communities is a way of doing dimension reduction. So rather than considering all of the nodes and edges in our network, we can group them together and consider just the community. So here's an example where we have 20 or 30 nodes and many connections, but once we reduce them to communities, we only have 5 objects to be concerned with. In applications, study of mesoscale structure in communities allow us to identify functionally related groups of nodes. Based on those partitions, we can also further diagnose the properties of nodes in the network as hubs that span modules and communities, or that might exert a particular amount of influence within their own community. The application of community detection and the study of communities, it can be carried out on both functional and structural connectivity equally well. And there's a lot of interest in how mesoscale structure and communities vary across individuals with respect to their disease cognitive states. But the real challenge is a technical one, right? If we have communities, we can do all of these things. But oftentimes when we encounter a network for the first time, we don't know its communities. We don't know its mesoscale structure. So how do we discover that? And it turns out this is actually a very difficult problem and one that we need computational resources to solve. So if we look at a network, here's an example of an adjacency matrix. The black dots indicate connections, and the white indicates the absence of connections. But it's not obvious if this network exhibits clear community structure or mesoscale structure. In fact, it has very cleanly delineated communities, but we wouldn't be able to tell that from visual inspection alone. So this is the same network, just its rows and columns have been reordered to highlight the communities. So in practice, we need to detect this type of structure algorithmically. And this is challenging for a lot of reasons. First, there are many different algorithms for detecting communities, and the user gets to choose them. These algorithms have different criteria for what constitutes a community. So the user has a big role in determining whether it's possible to find communities and the nature of those detected communities. And it turns out that in all practical cases, identifying the truly optimal set of communities is computationally intractable. It's not possible for even a modest sized network to consider all possible partitions and identify the best of them. The approaches that people use, the different algorithms, their results, the communities that they return to the user are sensitive to different parameterizations. Sometimes these parameters and implementations that people use, you're forced to select these parameters, but oftentimes you're not even aware that these parameters exist. And they're kind of just baked into the tool or the software. So today we're going to deal with this topic, detecting mesoscale and community structure in networks. And we're going to approach it, the rest of this talk, through the lens of the modularity quality function. I'm going to go through two examples, focusing first on structural connectivity, and then an extension to multi-layer networks, where we then study or go through an example where we apply it to a time-varying functional connectivity data set. And then we'll conclude with some additional readings. The aim of this talk is not to provide a definitive roadmap for community detection. Rather, community detection is user-defined. It's a flexible process. The goal here instead is to convince you that the community detection process is difficult, it's hard, and it's challenging. The user is forced to make many decisions along the way. And although oftentimes we want a clear roadmap, we want to say, do these 10 things and you'll have reasonable communities. The fact that we have to make these decisions along the way and the fact that there is no such roadmap, it actually makes community detection a very flexible process and allows it to be accommodating of particular and distinct research questions. So let's start with this first section. We'll deal with modularity maximization, modularity quality function, and we'll step through some of the sequences that we need to accomplish in order to apply to structural connectivity data. So it turns out and as I mentioned earlier, there's lots of different methods for detecting communities and networks. Some are very popular and used in network neuroscience, like InfoMap, which studies the probabilistic flow of random walkers moving through the network, and based on how certain subgraphs trap those walkers, it defines communities in a network. There's other community detection tools like clique percolation, which allow for overlapping communities. Similarly, methods like link communities may do something akin to that. Other statistical methods like stochastic block models try to fit models that explain the data, and among the parameters of those models are nodes' community affiliations. So all of these tools and many, many more exist in the network science and network neuroscience field. But arguably, the most successful and most popular is a tool referred to as modularity maximization. And you can see this is an outdated reference, but the original citation, original paper, cited well over 10,000 times. It's been around for it's had a long lifetime, and it continues to be used. And in modularity maximization, the communities that we're seeking, they're defined according to a very eminently simple principle. A community is a group of nodes whose actual connections is greater than what we would expect by chance. That's all. So to make this a little more concrete, imagine we have a network A. This is a structural connectivity matrix from a single subject. If we have some way then of determining what it means to be connected by chance, what the expected connectivity would be, which we can gain by running a null connectivity model, we can estimate what, on average, the connection should be between two regions under this model. That gives us our expected network, the weights of connections just by chance. And then modularity maximization simply takes the difference between those two. It takes what we see versus what we expect. And it gives us this new matrix, which we call B. It's the modularity matrix. And all modularity maximization would like to do is to put as many of those red elements, which are the ones that are stronger in the observed network than they are in the expected network, to put as many of those inside communities as possible, to choose our communities so that as many of those supra threshold connections fall within communities. So just to show you this intuitively, to build some intuition, the partition on the left, this is the modularity matrix, and here are 4 different communities. And you'll notice that within communities, there's lots of red. There's also lots of blue connections. There's connections that are greater than what we expect by chance, but also some that are weaker. So that's not a very good partition. On the other hand, the partition on the right, there's lots of red elements concentrated within the clusters, indicating that they are of high quality. They're taking edges that are greater than what we expect by chance and putting them inside the clusters. And I won't get in too much into the math, but it's worth seeing what this actually looks like in practice. So this is the modularity function, Q. So we detect communities by trying to make Q as big as possible. And so yes, as I said, this is the quality function. It's our objective function that we like to maximize. When we maximize that, we make it as big as possible. The hope is that we have good estimates of the network's communities. Aij is the observed weight of connections. Pij is the expected weight. And these parameters G, these are the communities to which each of the nodes has been assigned. And this last function is what we call the Kronecker delta function. And it's equal to 1 when 2 nodes are assigned to the same community. That is when Gi and Gj are equal, and it's 0 otherwise. So effectively, this summation is over all edges, over all nodes, ij. So it's effectively going over all edges. And it's only counting the ones that belong to the same community, and it's adding the difference in the observed and expected weight to this function Q. And in the end, we get, for a given partition, we get the Q value back. So the process seems simple, right? Our goal is to choose community assignments to make Q as big as possible. And there's lots of tools for doing this. There's lots of algorithms that can accomplish this, including agglomerative clustering, spectral clustering, simulated annealing. You name it, it's been applied to, or it's been used to optimize Q. By far, the most common and probably still most widely used is the Louvain algorithm. On benchmarks, it's proven to be both fast, so it has very low computational cost, but also accurate in the sense that it detects planted communities very well. I won't go into details about the algorithm, but just to give you a sense of how it works, it starts initially with every node belonging to its own community, and it gradually merges them. It goes through the nodes in a random order and tests whether moving 2 nodes and assigning them to the same community, merging their communities, yields an increase in Q. If it does, it accepts this change and it performs that merger. It does this until no individual node movements can improve Q further. You obtain something that looks like this. At this point, nodes have some community assignments. There's a green, a red, a blue, a lighter blue set of clusters. And then it performs what's called community aggregation. So rather than treating each of these nodes as its own node, they all get merged into a single community node, a meta node. And the process repeats. This constitutes the first pass, and then it does the same thing again and again, trying to merge meta nodes until they can no longer improve Q. So it's a very fast algorithm. It tends to be very accurate in practice. If you're using software like the Brain Connectivity Toolbox, this is implemented with the Community Lavaan algorithm. There's other software that's very nice for this, including Peter Muka and Mason Porter's version, the generalized Lavaan algorithm. They both implemented this style of algorithm for optimizing modularities. Okay. So how would we do this in practice? Well, if we wanted to now apply this to some real data, what would we have to do? So we have our network. This is that same structural connectivity matrix from our previous example. Just change the color map so it looks a little bit nicer. It's not just reds and whites any longer. We have our modularity quality functions. We have our network. We have a way of assessing whether a set of communities are good. And we have a tool, an algorithm that will optimize that quality function for us. So how do we then get communities? How do we get the clusters back? So here's our modularity equation again. As I said, we have our connectivity matrix. Our aim is to discover these communities, but there's this big kind of lingering question of this P variable, which constitutes the expected weight of connections, and it's generated, we obtain this value from a null connectivity model. So this is something that we have to choose, in fact. In most algorithms and most implementations, you're not actually given the opportunity to choose this. It defines in a very specific way, but in reality, it's a choice that we have to make, because that default a null connectivity model isn't always appropriate. And it's a non-trivial decision in the sense that it impacts the outcomes, and it impacts the communities that we detect by virtue of shifting which connections in the real network are expected and unexpected. If we choose different null connectivity models, some connections that have been previously stronger than expected now might be weaker, and so on. So we need to be careful about how we choose this null connectivity model. And I'll stress that there's probably not a singular correct model. It depends on your research question, context. It depends on the nature of the network that you're actually studying. So one really simple model would be to say, let's take the same network, let's just randomly scatter our connections, uniformly and randomly. This is akin to the Erdős-Rényi model. And if we were to repeat this an infinite number of times, the expected weight of every connection would be the same, and it would give us a matrix that looks like this, uniform. Possibly the most common null connectivity model is a so-called configuration or Neumann-Gervan model. And in this case, connections are formed randomly, but with the constraint that every node must have the same degree, so the same number of connections as in the real network, and same strength. And it gives rise to a slightly more constrained null model. And this is necessary in some cases, because we want to correct for nodes that make lots of connections. We don't want to put nodes into a community just because they have lots of connections. So this model accounts for that. And I said this is a very common null connectivity model, but it's particularly poorly suited for matrices that have negative weights, like correlation matrices, or where the edges have some statistical relationship with one another, just like correlation matrices, the weights in functional connectivity data. So this is a particularly poor or maybe not suitable model for those types of data. But of course, there's other versions too. We can try to incorporate spatial information into our null connectivity model with geometric random graphs or so-called gravity models. There's lots of ways of choosing this null connectivity model. But in this example, we're considering structural connectivity. The Neumann-Gervan degree preserving model is very common. So let's just consider that. For the sake of illustration, let's consider that particular model. So we've got our data, got our null connectivity model. We can calculate the expected weight of each connection, and we can push this into our Lavaan algorithm. So we run it one time, and it gives us this as our output. It gives us a Q value. It tells us how good a partition is, and it gives us the partition itself. So here, colors indicate the community to which a node was assigned. So for this first pass, we get a cyan, a blue, a yellow, and a red community. There's 4 communities in total. If we reorder our matrix, we see that yes, it seems to be doing a good job. Each of the clusters comprises a group of nodes that are internally cohesive. They have lots of connections to each other, but there's very few connections between communities. So we should be very happy with the outcome. But then, let's say you turned your computer off, or you exited out of whatever computing language you've been working in, and then you restart with a new random number generator with a new seed for your generator, and you run the algorithm again. And then, wait a minute, you get the same Q value or something very similar, but the partition is a little bit different. The actual cluster assignments are different. So you look again at the reordered connectivity matrix, and you're like, well, this still looks pretty decent. So what's going on here? I ran the algorithm the first time, I got one output. I ran it again the second time, and I got something different. What should I do here? Well, it turns out this is a consequence of two features. First, it's a consequence of the Levin algorithm that we're using. It's a non-deterministic algorithm. So if we run it many, many times, it's possible to get slightly different solutions each time. The outcome depends on the initial, how it was initialized. This is also a consequence of what we call the near degeneracy of the modularity landscape. So this is a consequence of the Q function. And it basically says that, imagine there is one optimal partition. There must be somewhere a slightly superior partition, the absolute best, the global max. But then imagine just flip-flopping the community assignments of one node at a time. Well, you're probably not going to get a really terrible partition, right? You'll get a partition that is still very, very similar to the globally optimal, but it'll be slightly different and slightly inferior. And you could do this if you have n nodes in your network. You could do this n times. You'd obtain n still probably very, very good partitions, but not optimal. So it turns out that with measures like modularity maximization or even InfoMap and lots of other objective function-based community detection tools, there are many near optimal solutions. And this can give rise to some variability in the outcome because we can almost never detect the truly optimal suite. So we're kind of sampling from on top of this plateau. We're sampling these very good partitions, but almost never the best. We get some variability at that plateau. There's not always the same solution. This is what I just said. Perturbations to good partitions give us other good partitions. Okay, so now this is, instead of just running the algorithm 2 times, suppose we run it 1,000 and we get, again, slightly variable results each time. So there's 1,000 different runs of the community detection algorithm. The rows are nodes and the columns are runs, and the color is the community in that run of that node. We can compute then a community allegiance matrix or a co-assignment probability matrix. So basically we ask for every pair of nodes, how often were they assigned to the same community? And if we saw all black and all white here, it would indicate that every single time nodes are either not assigned to the same community or they were. So it would be a perfect consensus. But of course, we see some gradations here. There are some nodes like these that were always assigned to the same community. They're bright white, bright yellow. Other nodes that were never, so the dark black, but sometimes there's these purple nodes. They were infrequently assigned to the same community. There's the red nodes that were maybe a little more frequently assigned to the same community, but not every single time. So there's some variability, and this community allegiance matrix demonstrates that. So how do we resolve this? How do we resolve this variability? We can do something called consensus clustering, and it amounts to effectively clustering the allegiance matrix directly. And we're going to combine consensus clustering with modularity maximization. And effectively, what we're going to look for then are consensus communities, groups of nodes that have higher than expected community allegiance, higher than expected co-assignment probabilities. So we take our observed allegiance matrix, and then we in some way permute the elements of this community matrix and obtain a an expected allegiance for every pair of nodes. And depending on how you do those permutations, if they're random and uniform, it becomes a flat probability, but you can use spin models that preserve some spatial information. But then we can compare our observed allegiance with our expected. It gives us a new kind of modularity matrix, and then rerun with this matrix our Louvain algorithm. And in practice, this will give us this will converge to a set of solutions. And so every single time you run the Louvain algorithm on our observed allegiance minus our expected, it gives you the same result. And so you can repeat this process until convergence, and it gives us and our consensus communities. And the consensus allegiance then will, of course, be binary. So you'll have nodes always assigned to the same community or never assigned with no middle ground. And in the brain connectivity toolbox, you can use the consensus undirected function to do this. Okay, so now maybe you're thinking, all right, we've resolved this issue. There's this variability in solutions is dealt with through the consensus clustering, so we must be done. Those are our new communities. Those are the right communities. Well, the answer is no, unfortunately. Modularity maximization and other methods like InfoMap have a so-called resolution limit. What this resolution limit means is that there are certain communities of certain sizes that even when they are unambiguously defined, I'll show you what I mean by that in a moment, the modularity function is incapable of detecting them. So here's an example of what I mean. So this is a network where it's a series of cliques, so maximally connected subgraphs that are connected to each other by just a single connection, right? Now, intuitively, if we would imagine if we were to give this network as input to modularity maximization, it ought to give us this. This is the expected solution, where every single clique is its own community. After all, these are densely, maximally connected internally, but very weakly connected to one another. But in reality, we get something that looks like this. We get these mergers of adjacent cliques. So why is this? What is happening? Well, it turns out that these, for networks of a certain size and density, the presence of even a single intermodular connection, so these connections that link cliques to one another, it may be so unexpectedly strong. Imagine you have a very, very sparse network, you just add one connection somewhere, but that connection is unanticipated by certain null models. That it's actually advantageous from the perspective of modularity maximization to group those two modules into a single cohesive community. And this is kind of a problem, right? It means that the modules that we detect by maximizing modularity, they may not necessarily reflect the network's true, or I'm going to say intrinsic, mesoscale or community structure. So this is a problem, right? We give our algorithm a solution or a network, and we expect a particular kind of solution back as output, but that solution can be misleading. So how do we address this problem? Well, one way to do it is to incorporate what we call a resolution parameter, a structural resolution parameter that allows us to manually tune the scale of communities that we detect either larger or smaller communities. So here is our original modularity maximization equation. And here I've added the resolution parameter gamma. So effectively what this parameter is doing is it's scaling the relative importance of the null model with respect to the observed network. So when gamma is really, really big, this null model counts for a lot. When gamma is really, really small, the null model counts for very little. And this has the effect of changing our modularity matrix. So when gamma is small, lots of connections are stronger than expected, and we'll get back very, very large communities. When gamma is very, very big, it changes the modularity matrix. So very, very few connections are stronger than expected, and the communities that we get back tend to be smaller. So here's an example of this. So in gamma is 0.2, we get a single partition. At 1, we get 4. With gamma of 1.8, I guess we have about 8, and gamma of 2.6, we get many, many more. And you can see the effect of varying gamma on a number of modules. So they increase monotonically as a function of gamma, And the module size also decreases monotonically as a function of gamma. Choosing gamma is particularly challenging task in and of itself. We've added a parameter, so in some ways we've made our task a little bit difficult, but we can do it many different ways. For instance, by trying to optimize some new heuristic quality function that we invent, possibly through cross-validation by comparing using the models that we have, the modules that we get for a particular gamma, imposing them on some networks that we've never seen, and choosing the scale based on whether we can maximize the gamma at which the unobserved data has a peak in its modularity, or by my preference is to embrace the multi-scale nature and recognize that brain networks are not defined, or at least they're communities at any particular scale, but rather we can consider communities of many different scales and embrace this multi-scale nature of community structure. And the last point I'll make with respect to this parameter is that this gamma of 1, which is the kind of the default setting, the one that if you don't mess with the parameters at all, this is what most implementations, most software packages use as their default parameter. It's not a very special solution in any sense. It's not, a gamma of 1 is not prioritized over a gamma of 0.5 or a gamma of 2. It happens to be one parameter value. So there's nothing special about it. You should always vary the parameter one way or another. Okay, so at this point we have communities. We have the ability to zoom in and out to find larger or smaller communities. What can we do with them? Well, one thing we can do is use the partitions, use the communities to assess node roles, for instance, by identifying hubs in our network. And we do this with two measures, the so-called within module degree z-score, it's a mouthful, that's shown here, and the participation coefficient. So what are these doing? Well, the within module degree z-score is measuring for every node, we're taking its strength of connections to its own community, taking the mean strength of connections over all the nodes assigned to that same community divided by the standard deviation. So effectively z-scoring a node's connections relative to all other nodes assigned to its community. So big values of z indicate nodes that relative to other nodes in the same community tend to make more connections. And then smaller and negative values indicate less, or fewer rather. The other measure is the so-called participation coefficient. And so what this is going to do is we're going to consider the weights, the strengths of connections from a particular node to any community, divided by its total strength or number of connections. And effectively what this is going to do, take the sum over all communities and then 1 minus this value, it's measuring whether a node's connections are concentrated within a particular module. If so, then P is very, very small and close to 0, or whether its connections are uniformly or highly distributed across modules, in which case its value would be close to 1. So it's telling us basically, does this node tend to form bridges between modules, or are its connections concentrated within? And so we can use these two variables within module degree and participation coefficient to kind of carve up a parameter space or a state space. So every node then could be situated within this space based on its participation coefficient or its within module degree z-score. And the space can be divided into hubs and peripheral nodes. So peripheral nodes tend to have very low within module degree z-scores. The hubs tend to have very, very high, so they make lots of connections or stronger connections relative to other nodes in their same module. And we can further classify them based on their participation coefficient. Hubs that have low participation, then those are nodes that have most of their connections concentrated within a single module or their own module. We call those provincial hubs. Other hubs span modules, we would call them connectors. And there's even these like hyper-connected hubs. We call them kinless, and we don't encounter them very often, at least don't describe them very often. And I'm illustrating here what I mean by those two types of hubs. So the connector hub, it links not only this module, but these three in total, whereas this provincial hub has lots of its connections concentrated within one module. So that's what we can do when we have modules and want to make statements about nodes and roles. So really quick summary. There are many tools and methods for studying community structure. Modularity is a function that tells us how good a particular partition is. It's a quality function. Intuitively, larger values of Q correspond to internally dense and externally sparse communities. And maximizing Q is computationally challenging. So we use heuristics, algorithms like the Lavon algorithm. This process, though, requires that we consider our choice of null connectivity model. It requires that we consider the non-deterministic nature of the Lavon algorithm, if that's indeed what we chose. We have to deal with variability over successive runs. And we also have to deal in some way with the near degeneracy of partitions. And again, this is something that we can deal with consensus clustering. And then finally, there's the issue of scale. So we have a parameter, the resolution parameter, that zooms in and zooms out at to detect different size modules and communities. Okay, so that was the first section. I want to now walk briefly through an extension of modularity maximization, and then we'll wrap up. And so in this extension, we're going to consider what we call multi-layer networks. So as we said before, modularity maximization is a very general framework, and it operates on a simple principle: compare what you see with what you expect. But the way we defined it previously was only for single layer networks, meaning when we have only a single adjacency matrix. There's lots of examples in neuroscience and psychology and the natural sciences where connection information, it's not restricted to a single matrix, but rather it's encoded across multiple layers. So I'll give you some examples. One possibility is with so-called time varying connectivity. So if we had a bold time series and we were to segment it into overlapping or non-overlapping windows, we might notice that over time, the connectivity pattern changes, right? So there's not a single connectivity matrix that summarizes the functional connectivity, but rather it's a sequence of time evolving networks. In this case, our layers would constitute the networks that we estimate at time 1, time 2, 3, and 4, and so on. So we have a multi-layer network where the layers are estimates of connectivity at different instants in time. There's lots of examples of this, and here are just a few. We can also consider layers corresponding to different connection modalities. For instance, we have the same set of nodes, their functional connections, their gene co-expression, a transcriptomic similarity, and their structural connectivity. These are three different modalities by which the same set of nodes can be connected. They're not really changing over time in the same way as the first example, but they can still be treated as layers in a multi-layer network. So layer 1 is the functional, layer 2 would be genetics, and layer 3 in this example would be structural. We can even consider cases where the layers correspond to different individuals, right? So we have an estimate of connectivity for one subject, for a second, for a third, for a fourth, and so on. So these examples, the way we can represent these data is not through single networks, because we have multiple networks, but rather we can represent them as a multi-layer network. This presents a challenge in terms of community detection. How do we study for a multi-layer network that has multiple connectivity modalities, individuals, or time points? How do we study its connectivity structure and its mesoscale structure? If we were to just average the layers together, we lose information. There's also decreased realism, right? By combining maybe different modalities, it really doesn't make sense to combine genetic structure and function and mush them all together into a single layer. But luckily, modularity maximization can rescue us. There is a solution here. And remember, all we need to make modularity maximization work is a modularity matrix, our observed minus our expected, and our optimization algorithm. So let's consider this example where we have 4 estimates of time varying connectivity. And I'll point out that this example, it could generalize to any of the other, any of the 3 that I presented on the previous page. Now we could, for each of these matrices, generate its own single layer modularity matrix, right? This network's its actual connections minus its expected. We use for all 4, just like we did previously with the structural connectivity. I'm going to present them here in this larger matrix for reasons that'll become clearer later. And we could, in this larger matrix, embed these matrices along its diagonal. And if we were to give this larger matrix to a modularity maximization algorithm, it would work just fine. And it would partition this layer, and this layer, and this layer. It would give us community assignments for each of the nodes in each of these layers. But there's a key, there's something missing here. And what's missing is that there's no links between the layers. So there's no connections between layer 1 and 2, 2 and 3, and 3, for any pair of layers. And what this means is that we will never ever merge a community found in layer 1 with the community found in layer 2. They'll always be separate, independent communities. This is because there's no advantage from the perspective of maximizing Q to group them together. There's no stronger than expected connections between those 2 layers. How do we resolve this? Well, we can add small amounts of interlayer coupling links from a node in a particular layer to the same node in a different layer. We can do it in many different ways. We can do it so that what we call a categorical coupling, so that node 1 is connected, and from layer 1 is connected to itself in layer 2, 3, and 4. Or we can do just nearest neighbor kind of coupling. So node 1 is connected to itself in layer 2, is connected to itself from 2 to 3, and then 3 to 4. But adding these links means that if we were now to give this matrix, this big matrix, to a modularity maximization algorithm, there's actually a possible advantage of grouping nodes from one layer and another into the same community. So that's actually very, very nice, because it allows us to directly compare the communities from one layer to another. And this also allows us to address questions, so when we're considering a time-varying network in this case, to identify stable or invariant communities, those that change from layers versus those that do not. And this is a measure that Danny Bassett and other people have defined. It's called the flexibility of a node. It's telling us how frequently a node's module assignment changes from one layer to another. And the process for doing this is fairly straightforward. And so we obtain the intra-layer modularity matrices, just like we talked about in the previous slide. It turns out that for functional networks with negative weights, it's a little more difficult. I won't go into details here, but it requires that we separately parse out contributions from positive and negative connections. There's many different ways to do this. This is probably the most popular and best known. But we can, for a signed and weighted network, generate a modularity matrix. So it's considering just the positive weights minus their expected and the negative weights minus their expected. We do it for every layer, layer 1, 2, 3, and 4. And then we get to add those interlayer connections. We have to decide, do we want to do it categorically or do we want to do it temporally, this nearest neighbors approach? In this case, because we're looking at time varying networks and there's some temporal precedence from one layer to the other, it probably makes sense to try to incorporate that into our multi-layer representation. So we're going to do the temporal version here, where we link layer 1 to 2, 2 to 3, and 3 to 4. But now we're confronted with something altogether new, and that is, how do we choose the weights of these interlayer connections? Or do we just add ones? Are they truly identity links, or should we weight them in some other way? And our choice of weight has important implications for the modules and communities that we detect. If we make the weights very, very weak, if we make these red interlayer links very, very weak, then the communities that we see in each layer will tend to emphasize the layer-specific communities. But if we make them much, much stronger, I'm indicating that here by the thicker lines, then the communities that we'll detect will reflect more closely the shared or pooled community structure from all of the layers. I'll note that this process has all of the same challenges as we saw with single layer modularity maximization. Right, we have to choose our null model. We have to deal with near degeneracy issues. We have to deal with resolution limits still. So there's a gamma parameter buried in here that I'm not talking about, in addition to choosing the interlayer links and their weights. So let's say we do this. We run our modularity maximization, choosing some kind of intermediate value for the weights of the interlayer links, and we obtain something that looks like this. So in this example, I've now added many, many more layers, I think 20 in total. So the rows represent nodes and the columns represent the layers. And if we want to identify then the nodes that are flexible, what we can do is we can compare successive layers and their communities to one another. Here is layer L and layer L plus 1. And look where these red points are. Those are nodes whose communities change from one layer to the next. We don't have to use any special similarity measures. There's no mutual information. There's no RAND index. It's literally just finding where these two have differing values for and in what elements. We do this for all consecutive layers. So here's, if we have 20 layers, and now there are 19 pairs that we're considering, and the red dots indicate nodes whose communities have changed from one layer to the next, and we can simply average those. So on average, how many times does a node's community or module assignment change from layer L to L plus 1? And this is the regional flexibility value that's been popularized in Danny Bassett's work and the work of others. We could project it onto the brain, and it gives us something that looks sort of like this. We can make it a little bit nicer if we repeat the algorithm many, many times, obtain many different results of flexibility, and now we get a little more continuous and smoother result. But that's the process in general for obtaining flexibility. And of course, the solution here, the pattern of flexibility, is going to depend a little bit on whether we have weak or strong or intermediate values for those interlayer coupling parameters. For instance, if we make it very, very weak, but then you'll notice there's less persistence of communities across time, across layers. But if we make it very, very strong, every layer tends to have basically the same communities with very little variation. Okay, so a quick summary. Modularity maximization can, of course, be extended to multi-layer networks. There's some additional considerations that we have to take into account, including how we define interlayer coupling and their weights. And it has all the same issues as before related to choice of null model, degeneracy, issues related to scale and resolution parameter. Okay, so I have one or two last slides related to some final comments and additional reading about community detection. So I should point out that there's important topics in the field of community detection that we didn't talk about at all here, that probably are of interest to many of you, including how to compare communities across individuals. We did not mention that another problem with Q is that entirely random networks, where connections are truly placed at random, can nonetheless have very, very high values of Q. And we didn't really get into details for choosing resolution parameters. So there's important topics in this space that just aren't covered here in these slides, given the amount of time we have. I'll point out that at the very beginning of these slides, I highlighted in the bottom right a citation to a recent book chapter that I wrote that does go through this in a little bit more detail. And you can also read the 2016 Sporns and Betzel review. It talks about this also in a little more detail. But there's some other bigger questions and points that I'd like to make as well. So modularity maximization is a very popular tool. So is InfoMap. These are probably the two most widely used community detection tools in network neuroscience. Both of them, however, have an internal model, implicitly an internal model for what kinds of communities they will detect. And specifically, they detect a class of communities that we call assortative, meaning that nodes in the same community tend to be connected to each other. This gives rise to the canonical internally dense and externally sparse communities. So you see this in the upper left-hand corner here. But there's other classes of community structures, other kinds of configurations that nodes can participate in besides this assortative structure. And they may be present in our data, but if we're only using modularity maximization and info map, we would never detect them. For instance, core periphery structure is another class of community structure where we have a densely connected core that projects weakly to a periphery, but the periphery doesn't connect to itself. We can also have the exact opposite of assortative communities, which is disassortative or multipartite, where nodes in the same community actively avoid connecting to each other. Instead, they project to different communities. Or we could even have a mixture where there's some different communities that interact assortatively, that interact as a core and periphery, and interact disassortatively. We would never ever detect core periphery, disassortative, or a mixture using modularity maximization or info map. To detect those classes of communities, we need different tools. And I'll advocate here for methods like the stochastic block model. There are flexible weighted versions of this, and we explored some of that here in our Nature Communications paper. Other people have done really great work in this space as well. So if you're interested in more general classes of communities, I would recommend seeking a different tool. And then the final point, which is kind of undercuts everything that I've said, it's that it turns out community detection is just a general enterprise. It is actually an ill-posed problem, and we can demonstrate conclusively that there is no singularly optimal solution for the true communities of a network, and we can specify the conditions under which no unique solution exists. So the way we can think about communities is that the communities that we observe, the lowercase g, some function of communities is what gives us our network, right? We have our network, capital G, of edges and vertices. We see this network, it has some communities. The way the links are configured in some way depend on communities. That's another way of stating this, right? And the goal of community detection then is to kind of invert this process, right? We start with the network, we start with the edges and the vertices, and we want to figure out what those communities are. It requires that we invert this F function, right? That's really what we would like to do. This presents a problem because it turns out that there may be many functions that give rise to a particular network configuration. So there's not a single unique function that gives rise to the network. And similarly, there might be multiple functions that give rise to the same network but have different communities. So it turns out this is a very difficult process to solve. And in essence, unless we know the true generating function, this true F that takes our communities and gives us our network, we actually can't unambiguously state that we've detected the network's true communities. We're estimating communities with modularity maximization or block models, but they're just that. It's an estimate based on a particular model, a particular expectation of what communities should look like. So this is a problem. It's something that we should always consider in our analysis of community structure. So, to conclude, my goal here was never to give you a definitive roadmap for detecting communities or modules in your network. Rather, I wanted to point out that it's a hard process. It requires that the user, you, make lots of decisions along the way, but ultimately these decisions allow you to incorporate details of your own research question into the community detection process. This includes choices of null models, parameterizations, and so on, which allows you to tackle questions that are of more interest to you rather than using some generic tool that can give you communities but might not be specific to your question. And as I stated before, there's a chapter out now that you can find on the archive. The book won't be out for a little bit longer, but it goes through some of these processes and spells out everything in the slides, but in much more detail. So this is where I will stop. And if you have questions, I'm looking forward to talking more in the chat. And hopefully you'll have some something we can talk about. I like talking about communities. Thank you.
Hey everyone, I'm Anne Blevins, and I'm excited to be here today to talk to you about multi-layer networks. So as you may have gleaned from the other excellent educational videos about network neuroscience, we have sort of a general process of analysis when performing these investigations. So what I mean is that we start out with our neurological data. Here I'm showing an example of perhaps some functional data. And then we do some sort of encoding process to represent it with our network formalism. And finally, the next step is to take that network representation and extract some sort of insight about our original system. Right, so this is, again, sort of a generalized pipeline, but I want to dive a little bit more deeply into this pipeline before we move on to the new material. And so this first step of encoding the data as a network is a pretty interesting one. So first, we're making a lot of decisions. I'm saying that I want to have a brain region correspond to a node in my network. So here I have 4 brain regions. I'll have 4 nodes in my network. Great. And then I'm making another decision about what edges are in my network. And here I'll say that 2 nodes will be connected by an edge if their corresponding brain regions are coactive at the same time in my scan. And I'm using an adjacency matrix to encode this network. We can represent this as either an adjacency matrix or network, but we actually perform later analyses with the matrix form of this, right? And in either case, we want to talk about our nodes and our edges and weights. So we can define our network based on the nodes, edges, and weights, or the adjacency matrix as well. The second step is the network analysis part. So I'm taking a network and I'm running some computation on it and getting out a number or a set of numbers or a set of labels or what have you. And that's not the important part. The important part is what that means for my system, right? I don't care if the centrality is 800, it matters what that actually means in the brain. So this second step is really about performing some computation on our representation that gives us actual insight into our original system. So let's now move to a little bit more of a complicated example where our data is actually from a recording of different tasks. So here we have the activity in task 1, task 2, task 3, and task 4. How should we represent this as like a computational object? One thing we might feel compelled to do is to create a set of networks where we have one network indicating coactivity in task 1, a second network indicating coactivity in task 2, and so on. Okay, so now we kind of have this bag of networks and we need to figure out how they fit together. All right, let's come back to that. Try another one. How about the case where we have functional and structural information? So here I have both the anatomical connections between regions and some functional information as well. Again, we're getting to a situation where we might try to make multiple networks. Alternatively, we could make one network in which we weight the edges based on some combination of their structural and functional connectivity. But either way, it sort of feels not complete, not necessarily the right tool for the job. Perhaps more complicated is when we have multiple parcellations. So here I have a nice case where in this kind of coarser parcellation, I have only this green node, but in the finer scale parcellation, that green node is broken up into 3 smaller ones. And again, we're getting into this case where we have a funky collection of networks and we want to extract, holistic insight of the whole system, but we have these multiple ways of measuring it. And finally, our last example comes from activity recording, in which we want to understand how regions correlate with one another across different frequency bands. And so all of these different scenarios from different data versus different questions are not represented well by simplex networks, is what we're called when we only have one network. Instead, they're much better represented by the framework of multi-layer networks. And so that's what I'm going to spend the majority of the time talking about today. What is a multi-layer network? And then we'll get into a little bit of how we actually use it. So first, what even is a multi-layer or multiplex network? And I'll tell you right now, the multiplex network is sort of what I consider to be the most natural extension or a natural starting point into multi-layer networks. It's a specific generalization or a specific case that we'll talk about in a little bit, but I think it's the best case to start with. So bear with me on that. So multiplex network is defined similarly to a simplex network, where we have a set of nodes, vertices, edges, weights, but then we also have this extra labeling function. So here, this labeling function takes every vertex. So here, there are 20 vertices, and it assigns a label to each of these vertices. And so the labels come from this label set L, in which L sub I is a particular aspect or dimension of the multiplex network. And so then we say that each vertex sort of is, as it corresponds to its label description. So what does that really mean? In this case, we have a very simple example where we have only 2 dimensions. This first one usually corresponds to what we might call the physical instantiation of the nodes. And so I've labeled them 1 to 10. And then in their second dimension, we have like different layers where we have A and B. And so for vertex 1, we would label it with 1 comma A because it gets 1 from this first dimension and A from the second dimension. Vertex 2 is 1 comma B. Vertex 3 is 2 comma A, and vertex 4, 2 comma B. And see how specifically based on how we've plotted this multi-layer network, that if vertices have the same label from dimension 1, they're plotted right above or beneath each other, indicating they sort of correspond to the same physical instantiation. And importantly, one thing that's very cool about multiplex or multi-layer network framework is that we can be very flexible about how we perceive the network. So even though in this case, I have colored by the second dimension, that gives us sort of one understanding of this whole object. I could just as easily color by the first dimension. And so there will be 2 vertices that, for every color, there will be 2 vertices, and we get 1 color for every element of this first dimension here. And so this will come up a little bit in the future, but it is something that I think is very unique to multi-layer networks, in that we get to have all these multiple perspectives, and the formalism can sort of handle that all at the same time. All right, so here we've talked about a multiplex network in which we have 2 layers, but we could have, far more, many layers even. So here we've got 5 different layers. Another way you might want to say it is that in the second aspect, we have, or second dimension, there are 5 different labels that a vertex could get. So we have, for example, 1A, 1B, 1C, so 1B, 1D, and 1E, and so on. This framework also incorporates temporal networks, where you can think about maybe a network evolving over time. And so here, again, this system is represented by the same nice mathematical formalism, where the second dimension here, the second aspect is now our time. It's our time points that we've measured the system. So vertex 1 has the labels 1, T0, vertex 2, 1, T1, and so on. And we can sort of track, again, this first physical unit across time. Okay, so again, I noted something about how multiplex networks were this nice case of multi-layer networks. So to be more specific, a multiplex network is a multi-layer network in which every node, and by that I mean every physical instantiation of this node, exists in every layer, and they connect, these nodes connect to themselves in each layer and only to themselves. And so when we see a visualization of this, we generally see a bunch of straight lines or vertical lines that connect nodes in different layers. So again, this might be the motor cortex, and this is the motor cortex here, and this vertex corresponds to the motor cortex in this different layer, and so on. But this doesn't have to be the case. Multi-layer networks are far more general than that. In particular, in the activity and in the study of node activity, perhaps we might want to study not just the holistic activity of nodes, but break it down into the different frequency bands and look at cross-frequency activity. And so here this was performed in a couple different papers, and they found that the hubs that were identified based on this multi-layer framework, in which they could understand correlations across frequency bands, were more predictive or better separated the schizophrenic versus healthy populations than hub information derived from a single layer or a simplex network alone. And additionally, these physical instantiations or these nodes don't even have to exist in every layer. So for example, in the parcellation case, here we only have 4 nodes, and here we have 10. So we don't have to have a perfect replication of these nodes across every layer. And here's another example. So this node exists in this layer, but not here in the orange one. But yes, again, it does exist in the green one, and so on. So these are very sort of general, it's the same framework, it's very general, can handle many different cases. For example, missing data would be a great one for this. Okay, so now that I've talked to you about multi-layer networks for a while, let's talk about what we can actually do with them. So again, we're returning to this sort of pipeline view of our network neuroscience. And so I want to start it again with this data to formalism arrow, talk about how we actually encode data into a multi-layer network in such a way that we can do something with it. So similarly to simplex networks who have adjacency matrices, a multi-layer network has a supra-adjacency matrix, in which the supra-adjacency matrix is similarly defined. We have a row and a column for every vertex, and the ijth element indicates the weight of the edge between vertex I and vertex j. And here I've colored by the layers in the second aspect. So we've got the blue and the green here. And so you can see on the diagonal, we have these blocks of intra-layer connections. So here's within the blue connections, and here's within the green layer connections. And then these off-diagonal blocks indicate the interlayer connections. And so here every node is connecting to itself in another layer. So we get these nice diagonals. And these can get quite large as, as our data grows. So here's just an example of the same super-adjacency matrix, but for this temporal network. And so we can see here how the intra-layer edges evolve across time. And importantly, this super adjacency matrix is one representation of this idea, and it helps this multi-layer network idea, but it's very useful because then we can perform, topological computations on this representation. Okay, so that was sort of how we take data and encode it as something we can really get our hands on. And now let's talk about this last arrow, how we take that, this encoding now, this representation, and derive some insight about our system. So similarly to the network case, there's a whole bunch of analysis techniques devoted to multi-layer networks or developed for multi-layer networks. Many tools developed for networks now have extensions to multi-layer networks, such as motifs, centrality, and community detection. But again, all of these are created so that we can, extract some insight about the original system. So all should be used with the original system and an original question in mind. For example, motifs in multi-layer networks. This is pretty cool. So let's say we have both the structural and functional information about the brain. In this 2017 paper, they looked at motifs within such a network, or such a multi-layer network, excuse me. And they found that indeed there's this overrepresentation of a structural and functional edge occurring between a certain pair of nodes. So that they can say that the structural edge is really informative about the functional edge and so on. But this sort of paired, one edge in one layer, one edge in another layer motif, it is one of the smaller motifs that we could look at. And they also expanded this analysis to larger motifs, and they can look at the representation of the different motifs within the network, and found that some were overrepresented than others. Additionally, we can calculate centrality in multi-layer networks, just similarly to, again, in the simplex network case. So for example, we could use random walks, but it's important to take into account that when we're calculating, or we're thinking about random walks in a multi-layer network, the walk can move across layers. So here, maybe we're in the green layer, and then we pop up here, and then we go over here. So there are many different ways that you can walk across this multi-layer network, which is why these centrality measures look very differently when calculated on nodes in a vertices in a multi-layer network, as opposed to those in a simplex network. Finally, there are also many ways that we can calculate community structure or derive community structure within a multi-layer network. So in general, the goal is to find collections of nodes that are tightly connected in all of the layers, or at least most of the layers. And in particular, this has been helpful in gaining a better understanding of the sort of independence of the visual and motor systems during learning. So looking forward, there are a lot of cool multi-layer network ideas on the horizon. It seems like a place that's just getting started. So in particular, as proposed in their 2018 review, we can use more than 2 dimensions. So all of the multi-layer networks that are shown in this presentation have had 2 aspects or 2 dimensions, usually brain regions and then something else, right? But we're not limited to that case. So here, in this example, we have brain regions. The second dimension is time, and the third dimension is subjects. This is a very common scenario for this field, where we collect not only the brain data for one person, but for many people. And with a multi-layer framework, we can incorporate all of that into one mathematical object. And furthermore, because it's all together, we can very naturally take all these really nice slices of the data, and it's sort of offering us a way to get multiple perspectives on the same larger object. So for example, I could pick just one instance in time and compare the networks across, or compare connections between brain regions across subjects. I could pick one subject and look at the connections between brain regions across time. I could pick one region and look at how it evolves over time across the different subjects. And so this is just highlighting, again, this sort of flexibility in subsetting and slicing this larger object. And again, it is akin to taking something that's really large and looking at it from multiple perspectives. Another place where multi-layer networks are really growing is in dynamics. So for example, here we had a system of Kuromoto oscillators in which each one was drawing from a pool of resources based on its activity. And so these authors looked at how the system evolved across time if they continue to pull from their resources, again, based on their activity. Additionally, dynamics, the set of authors looked at a system in which, or a multi-layer system in which the different systems interacted in different ways, either in interdependent or competitive or no interaction. And from this, they found that this dynamical system exhibited multiple interesting behaviors, including chaos and multi-stability and more. And so in particular, both of these studies aim to sort of answer this question about how processes spread and evolve across multi-layer networks and between layers, especially when there are both behaviors of the nodes themselves to consider and how the different layers interact. And so this is definitely an open problem, but something that has been very interesting and it's sort of just getting started. Additionally, another way we might expand the multi-layer framework is to incorporate pleiadic relations. So instead of having an edge, or instead of having a relation between nodes be restricted to an edge, so a dyadic relation in which only two edges or two nodes can be connected, we could expand to a sort of multi-layer hypergraph or a simplicial complex situation in which any number of edges any number of nodes can be related together either by a hyperedge or by a simplex. Finally, the area that's probably most near and dear to my heart is visualizing multi-layer networks. So this very cool recent paper that talked about the state of the art in multi-layer network visualization. And this is a very hard problem, but a very cool problem. And there are many examples of how one might go about this based on their data and their question. For example, all the visualizations I've shown you have been on multi-layer networks with very small number of vertices. But what if you have thousands or tens of thousands, right? How would you incorporate that into some reasonable visualization that fits on a page? So there are multiple strategies, including this interactive beehive, or a situation maybe where you only care about the interlayer connections. So each layer here is, so here basically each layer is a column and we've sort of collapsed out the intralayer connections. And then there are many other interesting strategies as well, perhaps coloring nodes coloring edges by layers and so on. So again, another problem that's probably not solved, but there are a lot of really cool tools at this point that can help based on of course, the data size, and the question being asked. So finally, before I wrap up, I just wanted to leave you with some examples of different code packages that can help you get started in analyzing, or encoding and analyzing multi-layer networks in Python, R, and in Julia. And then finally, I just want to say thank you to everybody who has helped out with this project and offered feedback in the presentation. And if you're interested in generating any of the images you've seen in this presentation, you can visit this GitHub repository, which contains code for you to generate any of the images on sites that had this little asterisk, or create your own multi-layer network visualization. So again, thank you for your time, and I'm excited to take any questions.
Hi, good morning. My name is Martijn van der Heuvel. Welcome to joining this lecture on multi-scale and multimodal connectomics as part of the human brain mapping course on connectomics. So the connectome, of course, describes this comprehensive map of connections of a nervous system. And in this lecture series, in this course, we have been talking about different technologies and different ways to reconstruct different forms of connectivity of the human brain. We've seen techniques like diffusion-weighted MRI and resting state fMRI to reconstruct the structural and functional connections of the human brain by means of neuroimaging techniques. And we have, of course, have also seen the importance of using network analysis tools to extract certain global properties of the reconstructed networks. And of course, with the link to find a connection between those connectivity architecture principles, like things like degree and short communication paths, modules, and how they relate, of course, to brain function and dysfunction. So far, we've mostly talked about technology and technical approaches and limitations and things to think about when you do network analysis on neuroimaging and other forms of connectome data. So in this lecture, we're going to shift gears a little bit and talk about some of the techniques and some of the concepts of how we can further integrate properties of the connectome with other types of neuroscience data. So throughout the lecture, I want to talk to you about multi-scale connectomics, right? The notion of that we can actually do connectomics and that we can study the connectome at multiple levels of organization. We can study it at the microscale level, in which we aim to reconstruct wide matter bundles by means of neuroimaging techniques. But in a similar way, of course, there is a huge amount of work going on studying connections between neurons, synapses, and also the global network organization of these cellular circuits. The second topic I want to talk to you throughout this lecture is so-called multimodal connectomics, the notion of the aim of integrating multiple modalities to get a better picture of the connectome in terms of its total organization. So for example, techniques and examples to integrate structural and functional data, or for that matter to integrate perhaps structural organization of the connectome or wide matter connectivity with microscale features like myelin measurements. So throughout the lecture, I will talk about multi-scale and multimodal connectomics not as two independent, completely separated fields, but actually they are very related to each other. Right? Most often when we talk about multi-scale connectomics, we most often integrate information from multiple modalities. If we aim to bridge the macroscale circuitry with microscale circuitry, well, most often we are indeed integrating neuroimaging data with cellular data. And the same holds for multimodal connectomics, right? Within multimodal connectomics, most often we are integrating multiple modalities, not just in one single scale, although this is of course possible, and we will see a couple of examples of this, but most often we are also integrating across scales. So throughout the lecture, I will talk about multi-scale and multimodal connectomics almost in one go, and I will explain several examples. So the connectome is the comprehensive map of all connections that together build a nervous system. Well, one thing we have to realize is, of course, is that in our field we are mostly inclined to study the connectome at the microscale level of organization, in which we aim to reconstruct the large-scale tracts by means of neuroimaging techniques. But the connectome and its network properties, of course, and its brain connectivity organization can be defined and, as such, examined at multiple levels. So as mentioned, in our field, we're mostly used to reconstructing the wide matter tracts and the system-level organization in terms of functional interactions by means of diffusion-weighted MRI and techniques like resting-state fMRI, reconstructing the large-scale structural and functional connections of the human brain. But of course, we have to realize that when we use diffusion imaging, for example, that we are indeed aiming to reconstruct the large-scale white matter bundles between brain regions, and that when we use resting-state fMRI, that we are really looking at coarse-scale, macro-scale, large-scale dynamical interactions between brain regions. And of course, once we have reconstructed these pathways at this macroscale level, we can then use techniques like network neuroscience to extract all sorts of features out of the reconstructed networks. And then we link all of these network features, or the aim we have is to link these network features, for example, to studying variation in connectivity in relationship to brain function and dysfunction in terms of disease, but also, for example, to examine how individual variation in network features might relate to things like cognitive behavior, or how they might change according to the course of development. But in a similar way, there is a huge parallel field in which, in a similar way, we can reconstruct the structural and potentially also the functional interactions of brain regions, not on this large core scale, but also on a more intermediate scale of level of resolution. Techniques like this, and examples of this are, for example, the mesoscale resolution connectome map built by the Allen Human Brain Institute of the mouse brain, in which they used fine-grained track tracing experiments to reconstruct at the mesoscale level, so in between the large-scale level that we aim to reconstruct with neuroimaging and the more fine-grained level at the single individual neuron level. So at this mesoscale, they were able to reconstruct the mouse connectome at this intermediate connection level. And interestingly, here too in this field, there is a strong growing interest of aiming to link perhaps the mesoscale level of connectivity and variation in mesoscale level connectome organization across individuals and across subjects, to link that variability in those connectome maps to, for example, variability in mouse strains. So here too, there is a suggestion that even at the mesoscale level, variation in connectivity might be related to also to variation in terms of brain function. And finally, of course, we can also even further go down and aim to reconstruct a level of connectivity at the neuronal level. Right here, of course, the aim is to reconstruct as good as we can the direct synaptical connections between neurons. So here, for large-scale nervous systems, that is, of course, extremely difficult to do, right? For the mammalian brain or the mouse brain or the human brain, reconstructing all synaptical connections, of course, for current technology is very challenging. But here, as an alternative approach, we can of course examine, for example, a certain column of the cortex and reconstruct all of the synaptical connections there. Our pioneers in this field are people like in the lab of Jeff Lichtman, for example, but also other institutes like the Allen Human Brain Institute are doing some amazing work in this field. Now, of course, for smaller nervous systems, there is the feasibility of reconstructing the full connectome map at this cellular, microscale, almost synaptical level of connectivity. Well, examples that we're all familiar of is, of course, the reconstruction of the C. elegans connectome performed in the 70s and the 80s, but still even today, people are aiming to provide even better and better, higher resolution reconstructions and more complete reconstructions of the entire nervous system of the C. elegans worm. And here too, there is a huge interest in studying these cellular networks, these microscale networks, by means of graph theoretical approaches, and aim to see how potentially underlying general architectural rules of connectivity might relate to the function of individual neurons, but potentially also to the behavior of the animal as a whole. Well, other examples are reconstructions that are on the way of the Drosophila fruit fly brain, around 100,000 neurons here. So you can already see the immense explosion of the complexity. But potentially also things like the reconstruction of, for example, the zebrafish larva, in which here too they used these highly advanced techniques to aim to build as good as they can wiring diagrams, connectome maps of synaptical single cell resolution connectivity maps, and see how whether here too principles of wiring might govern the overall organization of these nervous systems. Now, interestingly, here too, similar to what we have been very used to at our microscale level, there's a strong interest in the field of aiming to see, not just only to map the connectome of a certain species, like mapping the connectome of the sea ligan's worm or the zebrafish larva, but also aiming to see how variation in connectivity, potentially between individuals or between species, might relate to a variation in the end, functioning of the system, and in turn, of course, on the long end, in terms of variation in behavior. A good example of this is a beautiful paper by Boombager and colleagues a few years ago already, in which they reconstructed at the cellular level, so the synaptical connectivity level, a part of the connectome of the sea ligan's worm and a other worm species, the P. pacificus, and in which they showed that variation of connectivity at the neuronal synaptical level between the two species might also be related to variation and to differences in the behavioral repertoire of the two worm species. And in this case, it was the eating behavior. Well, I can highly recommend this paper because it is a very nice example of how not just only there's an aim of reconstructing a general connectome map of a certain species and studying that, studying the overall global wiring principles, but also how potentially variability in wiring principles and wiring organization might relate to variation in behavior. So this shows that at the microscale level, in which in our field, in the neuroimaging field, we are very interested in building the connectome and linking the connectome and variation across individuals, and link that to variation in cognition or variation in brain function, and perhaps making a contrast between healthy controls and cases in which we explore and how variation and changes in connectivity might relate to the formation of brain conditions like mental health disorders or neurological conditions. Also at the other levels, at the mesoscale level and the microscale level, there is a similar field in which here too they are interested in seeing how variation in wiring principles at different levels of connectivity might similarly be related to variability. So here too there are strong parallels between our field of the more macroscale connectomics and the field of mesoscale and microscale connectomics. So one interesting thing that we need to address here is that when people study the connectome at all of these different levels of organization, at the macroscale level using neuroimaging techniques like we do, but also at the mesoscale level as we've seen in the mouse brain, and for example also at the more macroscale, almost cellular synaptical level of connectivity in networks like the C. elegans, but also in terms of individual columns in, for example, the somatosensory system. One thing that people started to notice is then we can actually use the same network organizational tools that we are very used to examine the large-scale networks. In a similar way, of course, we can use these tools to also explore the organization of these cellular networks. And when we do so, is that there might be overlapping general principles of connectivity governing both the micro and the macroscale properties of brain connectivity. So there might be, in that sense, a large overlap in terms of the organizational principles. So one of the features that has been commonly reported is that both at the microscale and the macroscale level of organization, there is a strong tendency to form, or at least to have certain elements present that have a much higher connectivity than other elements in the system. People have noticed this at the neuronal level, but also at the terms of the mesoscale sub-area level in the mouse connectome, for example. And of course, we in the neuroimaging field are regularly reporting on the existence of hub regions in terms of highly association regions that are very densely connected and aim to integrate information. Or that's the theory, that these regions are important in integrating connectivity at both the structural and the functional level. So across multiple levels of organization, and even going further at the molecular level, but also at the even more macro scale level in terms of relationship between behavioral features, people have noticed the existence of these hub regions. And more general, we could argue that there might actually be certain organizational principles that are present all the way from the macro scale to the mesoscale to the macro scale, suggesting that certain principles of connectivity, whether it's the hub and the potential rich group organization, of the tendency to have the most highly connected elements of the network to be densely connected as a group, but also the tendency of, for example, showing a modular structure in which there is a strong tendency to have also segregation of information in the network. These are features that are present at both the macro scale and the macro scale, suggesting and fueling some theories that there might be underlying common principles of connectivity organization. And this, of course, also shows that if we examining the connectome at both the macro scale and the mesoscale and the macro scale, and examining organizational principles at each one of these levels can tell us a lot about potentially organizational principles at the other levels of organization. Examples of this are the notion of hubs to be first suggested to be present at the mesoscale, but then later on people also found these hubs to be present in neuroimaging data, and later on people also started to notice the presence of these hub systems, for example, and rich group organization at the cellular level in the C lipids. So here too, this shows that studying only one level of connectivity, one level of the connectome, and one level of organization at one scale might actually give us a relatively coarse view of connectome organization, and that aiming to study and potentially also integrate information across different scales of organization might be a very fruitful way to get a better understanding of how the connectome is organized, and of course how this connectome organization might be related to brain. So we could thus argue that the brain, of course, is this system that works at multiple levels of organization at the same time. And this is certainly not a new notion, right? There is, of course, a long-lasting understanding that the human brain indeed is this somewhat, which we could call a multi-skill system that operates at both the molecular level, at the cellular level, but also at the level of large-scale connections and interactions, at the system level, if you will. And of course, that all of these skills relate in their own terms to behavioral variation and might be a source of origin for the emergence of brain dysfunction and as such conditions of mental health and or neurological disorders. And over the last years, there's been a growing interest in not just only examining the brain at each one of these levels of organization, but of course also to search for potential interactions, cross-scale interactions from one skill to another skill. One of the most common that we're used to in the neuroimaging field, which we have already seen a couple of examples during this entire course, but also in this lecture specifically, in which we aim to link certain properties of the connectome, like wide matter tracts, or certain systems like the full mode network, to behavior, to variation in behavior. And we have also seen examples of how we can study how connectivity alterations might potentially be related to changes or mental conditions. Now, in a similar way, so this might be one level of interaction that we can study between different scales, right? Going from system level to behavioral level. But in a similar way, over the last years, there's been a growing interest also in the question then, how is the macro scale connectome actually related to features of the cellular organization of the brain? Or even further, how is it related to the molecular level or the genetic level of organization of our brain? So let's take a look at a few of these examples that have aimed to investigate these cross-scale principles, and also potential examples of how such cross-scale investigations might in turn hopefully get us a better understanding of how the the connectome at these different levels of organization altogether might form a foundation for behavior, and of course also might play a role in the emergence of certain conditions of mental health and or neurological disorders. Level of cross-scale investigations in the field of connectomics is, as we have seen, of course, linking variation and changes to connectivity at the system level to behavior and disease. Examples of this are beautiful work by Finn and colleagues of the group of Todd Constable, in which they adopt the term connectome fingerprinting in terms of how is individual variation in the connectome organization, how unique is that to a certain individual, and in particular, how might this individual connectome organization then also potentially be linked to variation in behavior. And in light of this, in a similar way, people have, of course, used extensively and are using extensively connectomics and the study of connectome organization in relationship to not just one condition, but also to multiple conditions, and seeing how potentially different functional systems might be related to different forms of mental health conditions. But these cross-scale investigations might also go a step further, and even going not just only linking connectome properties or wiring properties, connectivity properties of the brain to behavior and disease, but also aiming to see how certain connectivity principles, like connectivity organizational principles, might actually be paralleled across different levels, and how they might be related again to variation of behavior and disease. A good example here is an interesting paper by Ye and colleagues and Zalesky and colleagues recently published in Schizophrenia Bulletin, in which they actually compared different connectome organizations across patient populations with different levels of comorbidity networks. So here you can see that cross-scale investigation of connectome organization, in this case connectome organization at the brain level versus network organization of comorbidity of symptoms, might actually show parallel overlapping features, and that studying these organizational principles across these multiple levels of organization of the network, going from the system level to the behavioral level, might give us new insight into brain disorders. And in a similar way, we have performed some work in which we aim to compare cross-connectome or connectome alterations across a wide range of disorders, and actually started to coin the theory that potentially some certain connectome features, some features of the connectome and organizational principles of the connectome might also bring some sort of a general cross-scale vulnerability to a wide range of mental health conditions. So here too, these are examples of how investigations of organization of the network across multiple skills might provide us new insight into the emergence of potentially not one condition, but much more of a general vulnerability and much more general involvement of the connectome in a wide range of both neurological and psychiatric conditions. Now in this context, I think it is also important to emphasize not only the cross-scale or the multi-scale type of connectomic organization, but also emphasize a little bit more the multimodal type of connectome organization. Examples here are interesting work in which people aim to, for example, combine measurements of the structural connectome and the functional connectome, and also aiming to see how potentially the structural functional coupling, right, so the relationship between structural properties of the connectome and functional properties of the connectome might in itself be an important feature to be examined in psychiatric conditions. There has been several examples of reports of studies that have noted that the structural functional coupling, so the link between the brain structure and brain function, might be actually disorganized in various types of brain conditions. So one way to, of course, integrate information across different modalities would be to reconstruct both the structural connections and the functional connections of the brain and then linking them together. You can see a great example here of reconstructing a structural connectome and a functional connectome, and then of course looking at their interaction potentially at the edge level between the two forms of connectivity. There's also been other approaches being suggested. One of them is not just only linking structural and functional at the edge level, but also, for example, building networks at different levels of organization. This could be by means of structure, by means of T1, or function by means of resting state fMRI, but also integrating further even EEG and MEG and building a network of all of these different levels, and then combining these levels into a what is being called a multi-layer network or a multiplex network, in which network organization is defined at all of these different levels, and there is interaction possible between the different individual levels of organization. So here too, a more advanced integration of different modalities might provide us with additional information of how, and a more complex picture of how the connectome might play a role in again, brain function, and of course, in the end, brain dysfunction and disease. Other integrative models have suggested that we could also integrate different modalities and information of these different levels of organization, structure, and function in this case, not directly at the network level as a whole, but potentially more, and use our toolkit of network metrics, in this case the modularity organization, and perhaps combine at the level of network organizational features the different modalities. Right, in this case, the connectome was reconstructed by means of diffusion-weighted MRI, so the connections were defined purely based on anatomy, but the modular definition was defined on based off resting state fMRI modules, and this combining these two different forms of information, this study was able to show that hub nodes play an important role in functional integration in the network, and also that follow-up studies might have shown that this in this sense, this functional structural coupling might be disrupted in brain disorders. So these studies, I think, are interesting examples to show that integration of information can happen at different scales of organization of the connectome, but also at potentially within a single skill, combining different modalities operating at the same level of organization within that skill, but that further integration of these different modalities into a connectome model might bring us this more complete picture of how brain connectivity in the end plays an important role in shaping brain function and dysfunction in the human and more general in the animal brain. So a second level of cross-skill integration that is really worth to mention in the context of multi-skill and multimodal connectomics is a bunch of studies that have aimed to start linking the connectome level at the system level and the connectivity level that we are very used to when we study the mesoscale and macroscale connectome, all the way to the molecular and the cellular level of organization of the human and animal brain. Particular pioneering work in this field is being done by the group of Hilketak and colleagues, who have shown that variation across the cortex in terms of cytoarchitectural organization of cortical areas might actually be a very important constraint, or perhaps a shaping rule, if you will, for long-range connectivity between brain areas. But there's of course a long-lasting history and information known that different cortical areas of course have different levels of cytoarchitectural organization. Most famous example of course is the Brodmann atlas, which was defined on brain regions were defined in terms of their cytoarchitectural differences. So put differently, different areas of the brain of course have a different cellular composition, if you will, and a different layer structure, and this variation in layer structure, this cytoarchitecture, has been clearly linked to also for example, the integration capacity of particular brain areas. And it is thought that higher order association regions might have, in that sense, a completely different cellular architecture or cellular layout, a cytoarchitectonic layout that might make them enable them to perform and process information in a different way from, for example, sensory motor areas. And indeed, recent studies have also shown that this variation in cytoarchitecture and cytoarchitectonic class between different brain areas might actually be something that can also be measured, or at least is associated to specific type of advanced MRI metrics. So Heuvel and colleagues very elegantly show that if we take a look at these different classes of connectivity, of different classes of cytoarchitecture of the brain, that these different classes actually also have a strong tendency to shape the long-range connectivity between those areas. So their work showed that areas with the same cytoarchitectonic class also have a strong tendency to be more often connected to each other, clearly suggesting that there is a strong link between, on one end, how the brain is organized in terms of its cellular architecture, and on how long-range connections in the brain might be formed, and in that sense also how large-scale systems in the brain are being shaped. Further examples of this type of work, similar from the group of Heuvel and colleagues, a nice paper by Gulas and colleagues, who showed that indeed the cytoarchitectural similarity, so how brain regions tend to be similar in terms of their cytoarchitectonic organization, might be an important rule of wiring or principles of wiring of how long-range connections between brain areas might be shaped. This was observed both in the human brain but also in various animal models, so suggesting that this might actually be a much more of a common principle of organization in connectome organization. And going even a level further, there has also been a number of studies now suggesting that if we take a look at specific type of layer organization, for example, within certain areas of the brain, then that the cellular properties or the properties of certain specific type of cells, in this case layer 3 pyramidal cells, which are known to be important for indeed integrative capacity of certain brain areas, that these cellular properties, and in particular the variation, the differences between these cellular properties across different areas of the brain, might parallel variation in, for example, microscopic degree of these areas. This suggests that areas in which have on the macro scale a lot of connections and being connected across a wide areas and systems of the brain might also be brain areas in which the cellular architecture is tuned in such a way that these regions might indeed be able to integrate information across different sources. Now, of course, there is no strong causal relationship between the two, or at least we don't know what the causal relationship is. It could also be, of course, the other way around, that the cellular architecture of a certain brain region allows this area to form more long-range connections, and as such, form a much more central role in the overall macroscale connectome. But either way, right, whether it is A that is causing B or B causing A, in general, these studies are suggesting that there is a relationship between, on one end, the cellular architecture of the brain and differences between cellular organization between brain areas and the system-level, more macroscale organization of connectome and organization and functional interactions between brain regions. Now, importantly, there has also been a growing number of studies that have shown or start to show that this link between the cellular architecture, the microscale organization of the connectome, and the more macroscale system level of organization of the connectome might play an important role in shaping disease effects. There is fantastic work performed already a couple of years ago and still going strong, the work, of course, by the group of Bill Seeley, who pioneered the notion that atrophy patterns, as observed in terms of MRI atrophy, measured patterns of changes in brain organization in patients with different forms of neurodegenerative disorders, that actually these patterns might be related to the general system level of organization of the macroscale connectome. Right? Seeley and colleagues noticed that, for example, the pattern of atrophy in AD strongly tends to parallel the organization of functional connectivity in the default mode network, while, for example, behavioral FTD, the atrophy pattern there seems to be more overlapping to the pattern or the system level organization in terms of the salience network. And Buckner and colleagues showed for AD, in Alzheimer's, that tau and alpha beta deposition might actually potentially be related, or at least the extent to this deposition might be related to the level of functional connectivity of certain brain areas. So here too, they suggested that there might be a relationship on one end of changes at the cellular level, and at the other end, a change in connectivity, or at least a global pattern of connectivity as observed in the human connectome. And similarly, people have noticed that if we take a look at, for example, a disorder like schizophrenia, in which one of the key features that has been generally reported, one of the trademark features of the condition, is a change in layer 3 pyramidal neurons, and in particular reduction of spines, the number of spines, or a reduction in spine density of those neurons, in particular in frontal and temporal brain areas. And when linking this, or at least associating this to the change in microscale connectivity as measured by means of diffusion-weighted MRI, people have noticed a potential correlation between the two levels of organization. All right, suggesting that the regions that have the strongest reduction in spine density at the cellular level of organization might also be the areas in the brain that have the largest change in terms of their macroscale connectivity. So here too, this suggests that there might be a relationship between, on one end, the microscale connectome organization, and the other end, the macroscale connectome organization, and that this interplay might play an important role in shaping disease effects across multiple skills of organization of the human brain all at once. So one final level of interaction we definitely have to discuss in this context of this lecture is, of course, the interaction between, on one end, the system level of organization of our brain, and the other end, the genetic and molecular level of organization of our brain. And of course, in the end, to again provide insight into how this interaction might shape potentially brain function and brain dysfunction. Now, our first example of this type of cross-skill investigation is, of course, a recent growing interest in performing GWAS analysis on connectome and connectivity properties. And new resources like the UK Biobank, and of course also the Enigma Consortium, now allow, by means of large N studies, to perform these analyses. And this work is greatly pioneered by the group of Thompson and colleagues and Johansson and colleagues, who have done some first and brilliant work on performing GWAS analysis on connectome organizational principles. For example, this study elegantly showed that if we perform GWAS analysis on every connection of the brain, then there might be some genetic variance related to variation in connectivity properties of certain edges of the network, and that this genetic variation might also be enriched to genetic variation of, for example, Alzheimer's disease. So this shows, again, that integrating different skills and integrating information, in this case of the genetic level and the system level, and bringing these information sources together might learn us a lot about potential vulnerability factors for brain function and dysfunction. Well, a different approach, but I think in that sense very related, is not just linking genetic variation to connectome organization, but taking transcriptomic information and linking that to connectome organization. Well, here too, there have been several studies being performed and on their way of linking this variation across different areas in terms of gene expression data or protein expression data, and linking that to, for example, global organizational principles of the brain, and also connectivity properties. Well, Across a wide range of studies, studies have, for example, shown, and this is work done by the group of Fornito and colleagues, that if we take a look at the cellular level organization of the ceiling and the connectome, there we look at gene expression of the individual neurons, that for example, hub neurons, they tend to have a different genetic profile compared to more peripherally organized neurons. So this again suggests that there might be a direct relationship on one end between potentially genes and how genes become expressed, and protein levels for that matter, in certain neurons, and their capability, for example, to shape long-range connections and therefore to play potentially more central role in the overall connectome. Again, a direct interaction from one skill to another skill, and again, this might have a relationship between, for example, brain function and dysfunction. This study here showed, for example, that the expression of schizophrenia risk gene in certain areas of the brain might in that sense be related to the pattern that is also observed in terms of microscale connectivity changes in schizophrenia, with areas that have the highest level of connectivity change in the brain, these would be frontal and temporal brain regions, are also the areas in the brain where schizophrenia risk genes are actually the mostly expressed, or show the highest level of gene expression of these schizophrenia risk genes, suggesting that, again, there might be this important interaction from one skill to another skill, shaping forms of connectivity and disconnectivity. Now, as an important side note here, there has been, I think, important recent interest in also the methods on how we should integrate this transcriptomic data with neuroimaging data. So I can highly recommend here to read papers on spatial specificity models and gene specificity models, papers by Alexander Bloch and colleagues, and Markello and Misic, and also the interesting BIRD paper in Neuroimage very recently on the notion of proper statistical testing when performing this transcriptomic cross-scale neuroimaging research. And we have also put out a paper on also the need for not just only spatial specificity models, but also potentially gene specificity models to further provide insight into the strongest relationship on one end between the genetic level, the transcriptomic level, and the other end, properties of microscale brain organization. So taken together, in this lecture, we have talked about, on one end, both cross-scale connectome relationships and multi-scale connectome organization. The notion that the brain, of course, is not organized in one scale and one scale only, but actually that we have to realize that the brain is, of course, this system that operates at multiple levels at the same time. And importantly, one of the key topics of this lecture is that investigating the relationship between those skills and focusing on how one skill might be related to other skills and how they might be shaping, in the end, brain function and dysfunction might be an important new resource and a new avenue of increasing the complexity of the connectome and increasing this complexity in terms of how it might shape brain function and dysfunction. And importantly, along the way, we have seen a couple of examples of studies that have aimed to bridge, for example, the connectivity level and the system level all the way to the molecular and genetic level or the cellular level. And we've also seen approaches that, of course, can build bridges between the connectivity level, the system level, and behavior and potentially conditions of mental health and neurological conditions. Now, along the way, we've also seen that one of the interesting avenues to do so is not just only integrate information across scales, but also across modalities, right? That there is a huge benefit in not just only examining connectivity at the structural level and the functional level separately, but that integrating this information, for example, by means of multi-layer approaches or multiplex approaches, or by structural functional coupling approaches, might again provide us this much more richer view of the connectome and how structure might indeed potentially be shaping function and the other way around, and again, how this relates to brain function. Well, thank you very much for listening, and I hope you greatly enjoy the rest of the course and that you are inspired by the connectome field, and well, hopefully that you've also become inspired by integrating your connectome information across multiple scales and linking that to brain function and dysfunction. Thank you very much for listening.
Welcome to the lecture, The Virtual Brain. The brain works through interactions of its elements that are organized in networks at multiple scales. And it is impossible to simulate the brain at all levels of its organization due to, first, insufficient compute resources, and second, a lack of understanding of the rules or principles that govern the interactions. The virtual brain simulation platform provides a tool for a reduced, more abstract description of the brain network interactions using dynamical systems theory. And we will see how we can create a virtual avatar, a virtual brain of Jessica's brain using automated pipelines that process different types of imaging data. So here we reconstruct Jessica's head and cortical surface, the fibers that connect the different brain regions, the tractogram, and we parcellate the brain in different regions. And the regions are connected through the abstract anatomical skeleton. So the question is now, how can we bring Jessica's brain alive? So for this, we need dynamics, but we have already understood that it is impossible to simulate the brain with all its neurons. It's far too complex. So we have to reduce a model, and we do this in analogy to this flock of birds. As the birds in this flock, the neurons in the brain and many other interacting elements in the nature have the tendency to synchronize because they are interacting. So here, for example, you see a simulation of neurons. The space is spanned by the opening probability of the ion channels and the membrane potential, and the neurons are synchronizing. And this can be described through the mean field approximation. So the red cross in the middle is the mean field approximation that describes the average behavior of this population of neurons. And here you see a typical reduction process. We only keep the relevant parts, and this cartoon here shows the process of the reduction. So below that, you see the state space of circa 200 Hindmarsh-Rose neurons. So the Hindmarsh-Rose neurons are capable of bursting and chaotic behavior. And you see now in the state space that they build two dominant modes of behavior. And if we can describe these two modes, we capture the most relevant aspects of the dynamics of this network of interacting Hindmarsh-Rose neurons. So on the right, you see the equations of the reduced Hindmarsh-Rose model. So the original Hindmarsh-Rose model consists of 3 equations. Now we have a network of many interacting Hindmarsh-Rose models that organize in 2 dominant modes. And these are captured through these 6 equations. We have Xi, eta, tau that represents the excitatory population, and alpha, beta, gamma that represent the inhibitory population. Tau and gamma are slow variables. And then we have the local coupling parameters, K11 for self-excitation, K12 for inhibition to excitation coupling, and K21 for excitation to inhibition coupling. So here you see that the reduced version of the Hindmarsh-Rose model has the same qualitative behavior as the full spiking neural network model. So this allows us to now replace detailed spiking neural networks by mean field or population networks, as you can see here in the middle. Each brain region is represented by such a reduced population or mean field model, and they are interacting through the anatomical skeleton that often is derived from diffusion tensor imaging data and tractography. So in order to compute the dynamics that evolve through the interactions of the different nodes in the brain, we have the evolution equation that you see here. So basically, the activity in a population is a function of the preceding activity of this population, the short-range input and the long-range input. And the long-range input is mediated through the long fibers that can be determined through tractography. The short-range input is mediated through interactions on the horizontal level along the cortical surface, as you can see here. So the neighboring adjacent population influence directly this central population. This can be simulated, the local interaction can be simulated by local kernels, as you can see here. So here we have examples of a Gaussian, a Laplacian, and a Mexican head kernel that describe how the degree or strength of interaction is falling off with increasing distance. So basically each vertex here in this triangulation of the cortical surface contains one population, and they are interacting through these kernels. So the virtual brain allows to bring all these different ingredients together in a very convenient way. So we bring together the parcellation of the brain, the connectome, the surface geometry of the cortical surface and the head surface, and we can simulate neuronal activity, and then with forward models that are also contained in the simulation framework, EEG signals or MEG signals, and also fMRI bolt signals, functional MRI. The simulator has a graphical user interface and a command line interface. Here you see the simulator interface. We can choose between different local models. So here we have selected the generic 2D oscillator. There are many more models. We have looked in detail at the Stefanescu GSR 3D model. And we can also play with the parameters of the local model and the so-called phase plane viewer and see how changes of the parameter selection lead to changes of the dynamical repertoire of a population. This is a connectivity viewer where we can upload connectivities and display connectivities, but we also can construct artificial connectivities and put in lesions, for example, or alter the connection strengths between certain brain regions and simulate the impact on the evolving dynamics. We have also a stimulation cockpit where we can apply stimulation to brain regions. And here you see a simulation. You can visualize the simulations on the cortical surface, but we can also stick artificial virtual needle electrodes in the brain and probe the activity at the sites of the needle electrodes. So now in order to be able to simulate and reproduce empirical activity like resting state fMRI or EEG, we need to tune the parameters of the model. And for this, we choose 3 parameters, typically very few or only one parameter to avoid overfitting. And here we choose a global scaling factor G that scales the coupling between all regions. And we look at the ability to reproduce functional connectivity dynamics of fMRI data, and EEG features like bistability of the algorithm. We see here in this range of G, we have very good reproduction of empirical data. So now how can we learn and understand mechanisms of brain function using the virtual brain or brain network modeling in general? So here I'm showing a simulation of 22 minutes of resting state activity, fMRI activity, using the reduced Wang model. You see the equations here. We have input currents that drive an excitatory and inhibitory population. They're translated into firing rates of the excitatory and inhibitory population. And these are translated into synaptic activities of excitatory and inhibitory population. And by now, reproducing 22 minutes of resting state fMRI activity, we construct a model of interacting neuron populations that yield our observations, and at the same time give us for each individual brain region the time course of the firing rates of the excitatory and inhibitory population and the time course of the synaptic activities. And those we could not simply measure through non-invasive measurements. And here you see now in the upper panel our simulated data, and the lower panel our experimental data. And the simulated data of a single whole brain model span many timescales, reaching from a timescale of 100 milliseconds to a timescale of about 20 minutes. And here we have analyzed the state variables of our model with respect to certain features that have been investigated in empirical data in the monkey, in the rat, or in the human. And we can see that a single full brain model encapsulates many observations that come from even different species and different experimental methods. So for example, here in the very fast time window of 100 milliseconds, we see the relationship between firing rates and the phase of a single alpha oscillation. And we see that at the peak of the alpha oscillation, there's a trough of the firing rates. And the same is observed in real experimental monkey recordings. And here on the right, we see the switching of the functional connectivity over time, the functional connectivity dynamics in the simulated data and the empirical data. So now that we have such optimized full brain models in the study for 15 healthy adults, we can probe how injected activity is processed in the brain, whether this corresponds to the observations from experiments. So what you see here are grand averages of the state variables, the input currents from the global network, the local excitatory input currents, and the feedback inhibitory input currents that sum up and drive the excitatory and inhibitory population, the resulting firing rates and synaptic activity. And the state variables are averaged according to the zero crossing of the alpha cycle. So now we can follow the summed up activities through the excitatory and inhibitory population. The axes indicate the amplitudes that go, the right axis in the inhibitory population, and the left axis, the amplitudes that go in the excitatory population. And we see here the input currents, the summed up input current consisting of feedback inhibition and local excitation that drives the excitatory population and leads to the firing rates of the inhibitory population that then are inverted because of the inverting effect of inhibitory populations, and then summed up with the local excitation and global network excitation. So these three input currents sum up and drive the excitatory population, and due to the dominance of the inhibitory feedback current over the local excitation and global excitation, the summed up input current that drives the excitatory population follows closely the shape of the feedback inhibitory signal. And this leads to a firing rate that has its minimum at the peak of the alpha cycle. So due to the feedback, the dominant feedback inhibition, we see a pulsed inhibition of the firing rate of the excitatory population, and consequently also pulsed inhibition of the synaptic activity. So our model shows us how the incoming alpha cycle leads to a pulsed inhibition of the firing rate of the excitatory population. And this is exactly what we also observe in empirical data. So here on the left, we see our simulation we see the firing rate in relation to the alpha cycle, and we see that at the peak of the alpha cycle, we have minimum firing rates. And here on the right, you see experimental data from monkey recordings. So now in mouse and rat recordings, we can measure inhibitory postsynaptic potentials and excitatory postsynaptic potentials. And it has been shown many times that excitatory postsynaptic potentials are paralleled by inhibitory postsynaptic potentials, exactly as is the case in our model. So we now look at the experimental relationships between the amplitudes of inhibitory and excitatory postsynaptic currents. We see that here the inhibitory postsynaptic currents are 5 times larger in amplitude than the excitatory, and this is the case in our simulations. And we also see that the inhibitory current follows a few milliseconds after the excitatory current. And this is also the case in our simulations, where the excitatory populations feed the inhibitory populations, and then we have a slightly delayed feedback inhibition that goes back in the excitatory population. So now we zoom out a little bit and we look at the effect of an artificially injected alpha spindle. So if we inject such an alpha spindle, the orange envelope shows the increase of the power, of the alpha power in our system. We see how the inhibitory population leads to a rectification of this alpha spindle. The negative deflections are filtered out because there's no negative firing rate. Then this rectified signal is inverted. We have again a dominance of the feedback inhibition over the local excitation and global network input that drives the excitatory population and leads to a drop of the firing rate. So if we have an increase of alpha power, this is resulting in a drop of the firing rate, drop of synaptic activity, and here at a slower time scale to a decrease of the fMRI signal. So this explains mechanistically the observations from simultaneous EEG fMRI of an anti-correlation of the fMRI build signal and the EEG alpha power. And here again, we see our simulation output, the relation between the firing rate and the alpha power. It's an inverse relation. And similar observations have been done in experiments with monkeys. And not only the firing rate is negatively correlated with the alpha power, but also the behavior. So the lower the firing rate and the higher the alpha power, the worse is the discrimination ability of the monkey in a somatosensory discrimination task. This means that the dynamics that we are observing and capturing with our model are also tightly related to the cognitive function. So now we zoom out even a step further and we inject slowly modulated alpha activity with different cycle frequencies of 0.01 and 0.03 Hertz, indicated by this orange envelope again. And we follow this signal through our excitatory and inhibitory population. We see again a rectification of the signal in the inhibitory population, inversion, dominance when driving the excitatory population, and to a pulsed inhibition again of the firing rate, synaptic activity, and fMRI that leads to an inverse relation between the fMRI signal and the alpha power, as we observe in experiments. But interestingly, the slow modulations of the alpha power with different cycle frequencies also lead to differential amplitude modulations. The slower the cycle frequency, the higher the amplitude modulations. And if we look closely, we see that this amplitude modulation emerges in the brain network. So there's an accumulation of excitatory activity if we have more time for accumulation, if the cycle frequency is longer. If we now turn off the large-scale network, we see that this frequency amplitude relationship disappears. So it requires the global network in order to have a frequency amplitude relationship that is also responsible for the scale-free behavior in our resting state data. Here you see the power spectral densities for empirical fMRI, for our simulated fMRI with the global network turned on. And if we turn it off, we see a flattening of the power spectral density of the beta exponent. So now if we look at the parameter space and the different features that we capture with our simulations, raw fMRI, time series, resting state networks, functional connectivity, and the power law exponent indicating scale freeness. The parameter space here is spanned by the large scale coupling factor G, so large scale excitation, and feedback inhibition J. We see that there's a single subspace, this diagonal, for all 4 different empirical features where we have best predictions of these empirical data. So it needs a proper balance between large scale excitation and feedback inhibition in order to make the different biological plausible behaviors appear. And here you see the simulation again with all its features. So it's a single model that captures many different timescales and behaviors in a single theory of the brain and allows us to understand the rules or mechanisms that link the different observations together. So I hope I have convinced you that we can learn and better understand the principles of brain function. But what makes us human is not the dynamics of our brain, but the resulting function, and in particular the intelligence. And this is also an aspect that we can explore with brain simulation. Here, this is unpublished work. We plug in circuit models that are capable of working memory and decision making in our brain network models. We stimulate the circuit models with ambiguous motion dot stimuli, so evidence and a lot of noise go in these 2 competing populations, A and B, that are mutually inhibiting. Now we simulate the brains of circa 750 individuals. And interestingly, the decision performance, the simulated decision performance, depends on the fluid intelligence of the virtual brain owner. So the same plug-in circuit models perform differently in different brain network models. So this is a very interesting result because it enables us to explore the mechanism behind this differential behavior. So what you see here are the state space spanned by the 2 competing populations, A and B, for 4 different scenarios. Different scenarios for different degrees of background correlation or functional connectivity indicated by R, 0, 0.5, 0.9, and 1, and the fluid intelligence. So higher functional correlation, connectivity is associated with higher fluid intelligence. And we see here 1,000 simulations with the gray lines for each scenario. And the simulations are averaged according to the decision, either for population A or population B, indicated by the orange lines. And as long as the simulation, the activity of population A and B goes along this red diagonal, they are in a balanced state. If it deviates from the diagonal, either population A or population B wins. But if we now look closely, we see that the higher the correlation in the network and the higher the fluid intelligence, the longer population A and B stay in a balanced state. And this means that they have more time to accumulate the evidence and average out the noise, which leads to better and more correct decisions. So in this way, we can understand the mechanisms underlying even cognitive function on the basis of brain network modeling. But we can also use brain network modeling to explore clinical diseases and develop novel therapies and diagnosis tools. So here you see a list of different studies using the virtual brain simulator or brain network modeling in the context of epilepsy, brain tumors, genetic variants, stroke, and dementia. Several more are in the pipeline. And I want to give an example for dementia. Here we see the distribution of positron emission tomography measured beta amyloid plaques. And we know from cellular and animal studies that in the vicinity of these plaques that you find particularly in patients with Alzheimer's disease, the function of inhibitory interneurons is impaired, which leads to disinhibition of excitatory interneurons and to hyperexcitation. And this cascade we can model in our local models. We can map the burden of the beta amyloid on a region-wise level to each individual population model. Here we choose another population model, the Janssen-Ritt model, consisting of a mean field approximation of the pyramidal cells, inhibitory interneurons, and excitatory interneurons. And the beta amyloid burden changes the coupling between the inhibitory interneurons and the excitatory pyramidal cells. And here we can now study with our mathematical tools the implications. So we see here the activity of the pyramidal cells and here of the inhibitory interneurons as a function of the network input that you see here, the global network input. And we see that depending on the global network input from the brain in which the population is embedded, the dynamic repertoire changes. Here we have a limit cycle, another limit cycle, and here we have a fixed point. So now if we change the beta amyloid burden, which means the coupling between the inhibitory interneurons and the pyramidal cells, indicated by this coupling parameter J, we see that the dynamic repertoire of the population changes. So with each increase of parameter J, we see a change of the geometry in the state space. The limit cycle size changes, and also the location of the fixed points. And if we look now at the resulting simulated local field potentials, we see a slowing of the local field potentials. And this explains nicely how the accumulation of beta amyloid in the brain, so protein, leads to altered dynamics in the brain in Alzheimer's disease. So this is just another example how we can better understand the mechanisms not only of brain function, but also of brain dysfunction. So now one limitation, we are opposed with the virtual brain as a mean field approximation. So everything below that scale, we are simply missing. If there's plasticity on a single neuron scale, we will not capture this with a mean field approximation. And for some mechanisms or processes in the brain, like learning, we will need to also take into account these detailed processes. So for this purpose, we have developed co-simulations that allows to simulate some selected regions in the brain at more detail, for example, as spiking neural networks. And as a first application, we used already existing implementations of spiking neural networks of the basal ganglia and plugged these existing spiking neural networks of the basal ganglia and full brain virtual brains. The purpose was to identify an approach for in silico planning of deep brain stimulation for patients with motor disease. The new European infrastructure eBrains has been created by the Human Brain Project, the EU flagship project. And this eBrains infrastructure offers services for neuroscience, including data and knowledge services, digital atlases, simulation services, and so forth. And we have deployed our services around the virtual brain on eBrains. We have connected our services that you see here in the blue column with the core services of eBrains, the knowledge graph that provides the possibility to ingest data and make data discoverable, and the high-performance computing backend that provides the computing resources that are required for processing of complex data and for the simulations. Another feature is interface to the digital atlases that allows to draw directly information from the digital atlases in the brain network models to constrain the parameters of our models. And with this, I would like to end my presentation. I hope you got some interest in the brain network modeling approach. And if you like, you can go directly to eBrains and start the virtual brain simulator in the cloud. Thank you very much for your attention.